Good morning, Good morning, sir. Today's lecture will be delivered by Professor Bishnab Charan Tripathi, sir, popularly known as BC Tripathi, sir, from Sharada University, Uttar Pradesh, Noida. And his areas of research and interests are in molecular plant physiology, photobiochemistry, and biotechnology. He significantly contributed to our knowledge in photosynthesis. His work on 5-amino linoleic acid, a precursor to heme of chlorophyll, as a photosynthetic herbicide, and his discovery of suppressing photomorphogenesis by red light is mediated by phytochrome A is a novel and interesting area of research. And his education, he has completed his PhD from JNU, Jawaharlal Nehru University, from New Delhi. And his postdoc, he has completed from University of Illinois, Urbana-Champaign. And his publications were in several reputed journals like Plant Physiology. Most of us, our lab papers will be published in Plant Physiology, and Plant Cell and Environment, Journal of Biological Chemistry, Photosynthetic Research, and Plant Biochemistry, Journal, and Plant and Cell Physiology, and more. He also contributed many researchers in rice also. He recently has given last year genotype to our lab to proliferate the number of genes and number of plants and give it to us to him. Like he has given a very good opportunity for us to learn about Poriza sativa indica, which he has developed. And he also has some many honors and awards also. He was elected as INSA Fellow, which is also known as in Indian National Science Academy, and he is also a fellow of National Academy of Science and National Academy of Agriculture Sciences, and he is also a recipient of a J.C. Bose Award and National Fellowship of Government of India. He received NASA or NRC Senior Fellowship and Kennedy Space Center, USA, and recipient of Rockefeller Foundation Biotechnological Career Award, USA. And here we all warmly welcome our today's guest of honor, BC Tripathi, sir. Once again, welcome, sir.
Okay, thank you. Thank you. You can sit and that's okay. I can sit and talk both. I'll do that, no problem. Okay, good morning to you all. Is okay, I'm audible to all of you? Okay. You don't need mic. Yeah. So, I'm sure all of you by now you know a lot about photosynthesis, chloroplast biology and all that. And the mechanism of photosynthetic details and all that, right? Light reaction, dark reaction, right? So-called light dark reaction, that is light independent reactions and all that, right? So the basic unit, basic local organelle, where all these take place, is chloroplast. All of you know that, right? And when you talk about chloroplast, it is today's chloroplast. Chloroplasts are not there when it evolved, okay, before it evolved something else. There are Bruggen-Gargi, cyanobacteria, right, who became endosymbiont in amoeba-like organisms, right, and after billions of years became a chloroplast, right. So what happened when a free-living organism, okay, invades a animal cell or a non-photosynthetic cell, okay, that is like cyanobacteria, Bruggen-Gargi. Then you see, many things, okay. Let's say a free-living organism, does not need genes for osmoregulation. because already they are in the host. So you depend upon a host now, right? So many genes phosphorregulations were out, lost from the what? From the endosymbiont. We call it endosymbiont or cyanobacteria, right? Endosymbiont. So many genes were lost. Many genes were also transferred from the ancestor, that is cyanobacteria, we call ancestor, to the host cell, nucleus, right? It's a eukaryotic cell, which then got a prokaryotic system, right? So many genes are transferred, and some genes still remained inside the chloroplast. Okay. All genes are not transferred. Then there are basic reasons why all genes are not transferred, so to the nuclear genome, became a chloroplast. Okay, you see, when cell division takes place, if you've seen, okay, nucleus divides, at the same time, chloroplast also divides. Not it? Chloroplast divides into two. One chloroplast divided into chloroplast. Or how the chloroplast is going to multiply. Okay. And remember, each chloroplast have 30 to 200 DNA, circular DNA, right? They are circular DNA, right? Because cyanobacteria is a circular DNA. They are all inheriting the cyanobacterial system, right? Prokaryotic system. So they have also circular DNA, chloroplast. They have 70S ribosomes, chloroplast 70S ribosomes, right? And all the translation making machinery or transmission machinery are prokaryotic in nature inside the chloroplast. Okay? So a prokaryotic machinery is surviving inside the eukaryotic machinery, right? You see that? And this survival depends upon the mutual interaction of chloroplast genome and nuclear genome. Okay. So nucleus, of course, takes over the central command of chloroplast, right? For its multiplication, for its proteins. It's a lot. You see, if you see chloroplast proteins, you know what you have. 95% of the proteins or genes are transferred from the ancestor or Brugina alga into the nucleus. Only less than 5% of the genes are remaining in the chloroplast. All right. So, although the chloroplast can synthesize 5% of its own protein, still 95% of the proteins are synthesized where? In the cytoplasm by the nuclear genome on 80S ribosomes and post-translationally imported into chloroplasts. Okay. Now, that's the way the chloroplast exerts a command over the chloroplast. Okay. But chloroplast has its own genome, right? As I told you, only 5% of the proteins or less than 5% proteins are coded by the chloroplast genome. So, the chloroplast also sends a signal to the nucleus. It's not one-way traffic. Nucleus sends signal to the chloroplast, and chloroplast sends a signal to the nucleus. The mutual, okay. And what that signal is? Okay, you see, let's say the chloroplast is under stress. Let's say, and let's say it has lost a lot of chlorophylls for some reason. Okay, maybe under any, let's say, developmental stress. When you talk about, then you can talk about abiotic stress, okay, or biotic stress, okay. Then what happens? Chlorophyll biosynthesis is reduced, right? Chlorophyll biosynthesis becomes minimal. And chlorophyll binds to where? All of you know by now, so I'm not going to details of that. Chlorophyll binds to what? Light harvesting chlorophyll protein complex. A light harvesting chlorophyll protein complex coded by the nuclear genome. Not it. Not by chloroplast genome. Okay. So you don't want a scenario where the chloroplasts have, let's say, 20% of chlorophylls, but nucleus is synthesizing all 100% of light harvesting chlorophyll protein complex. okay, as usual. Okay, so nuclear gene has to shut down. Okay, or has to gene expression is reduced. So chloroplast gives a signal to the nucleus saying that I have now less chlorophyll with me. I have now 20% chlorophyll with me, not 100%. So it gives a command to nucleus saying that don't synthesize so much light harvesting chlorophyll for me anymore. Okay, because I don't have that much chlorophyll to bind with it. Okay, all right. So therefore, the signal that goes from the chloroplast to the nucleus is called retrograde signaling. All of you might have known about it. If you don't know, you must know by now the retrograde signaling. That chloroplast could give a signal to the nucleus also. Okay, so the traffic is mutual. Okay, and one interesting thing is also see there are only one or two organisms which evolved long ago, but much later than endosymbiont. Okay, so these organisms are somehow intermediate. In the sense, they are engulfed cyanobacteria. It's not alpha cyanobacteria, beta cyanobacteria. They engulf the cyanobacteria, different kind of cyanobacteria. Same, still they are available in nature. And these cyanobacteria became an endosymbiont, but not complete endosymbiont yet. Okay, all right. This happened around 50 millions of years ago, or 80 millions of years ago, compared to the eukaryotic alga that started 1.5 billions of years ago. Okay, 1.5 billions years ago, we believe chloroplast started, became endosymbiont. Okay, and here there is this other species I'm talking about. Okay, this species only engulfed a different kind of cyanobacteria, but it is still chloroplast is making. There are some chloroplast features are there, okay, but not everything is lost from its genome. Some genes are gone, but still its protein import machinery, is not through import apparatus, but through vesicular transport, saying that it still has some prokaryotic features, okay, and many other features. And its chloroplast DNA is much bigger in size compared to the nuclear DNA of other chloroplasts we see in nature. So in nutshell, this gives an introduction of origin of chloroplasts, right? And we'll not talk in details of this procedure, okay? But if you do not know, just have to remember about this. And the chloroplasts also can, as I told, divide itself, okay? Become 1 to 2, 2 to 4, and so on and so forth, okay? And otherwise, when cell divides, chloroplast will divide, chloroplast multiply. Can you say a chloroplast is a plant which single cell plant and have millions of chloroplasts? Can you call it algae? The algae, acetabularia. Okay, 5 centimeter, one single cell is 5 centimeter long. Okay, nucleus is the bottom. Okay, and whole 5 centimeter circular cytoplasm have millions of chloroplasts. Okay, and this is regulated by one single chloroplast called one single nucleus. Think about that. Nature, diversity is so much, okay, in the process of evolution. And it's not polyploidous, it's a diploid plant. Now, this is haploid plant. Algae is haploid plant. Okay. So let us start a little bit details, of course. Yeah. So as you say, it's a photosynthesis. The oxygenating phototrophs, right? Having photosynthetic apparatus are determined to occur evolution on Earth. What relation between proplastids, plastids and chloroplasts, we'll talk about a little bit more. In this talk, a little bit, not much. What do you know about the size, shape, and number of the size of the organelles? All of these chloroplasts, all have seen the structure, have this thylakoid membrane here. Okay. All right. Thylakoid membrane structure from the granum. Okay. All of you know about chloroplasts, right? Outer membrane, inner membrane, intermembrane space. Sorry. Intermembrane space. These have thylakoid membranes formed stacked from the granum, right? All of you know that, okay? They're discs, right? So if you see from the side, they are stacked. But if you see from the top view, you see from the top, so you stack like that. But you see from the other side, you see the real structure, right? Stacked from the granum. Okay. So, is there, making 50% of total protein of the plant actually. Rubisco, right? Rubisco constitutes 50% of the protein. Okay. So, why do you think Rubisco is so a big fruit, so much? You see, plant is investing 50% of its amino acids on one protein called Rubisco. Right? Right? One protein. And the rest 50% amino acids are used in all other functions. In the respiration, nitrogen assimilation, gene regulation, gene expression, you name it. Okay. Stress tolerance and all that. Okay. So because Rubisco is so vital for this plant survival. And Rubisco is a sluggish enzyme. Okay. It works slow. Okay, its turnover number is very low. Okay, therefore, you know, it is a fact, you know, that we need more Rubisco for the plant to survive. You see, plants are photosynthesizing and also respiring. Okay, you know it. Chloroplast is photosynthesizing, and who is respiring? Mitochondria. Yes, for respiration. Okay. And do you think this, if the rate of photosynthesis and respiration is same, or more, or what? Photosynthesis is more, respiration is more. And what? The rate of respiration is much lower than the rate of photosynthesis. Much lower. Because respiration taking place whole time, during day and night. Photosynthesis taking place only during daytime. Not only that, unless the photosynthesis is much higher than respiration, then how the plant will grow? Plant becomes zero. Right? Plant will not grow. Carbohydrate accumulation will not be there. The rate of carbohydrate formation equivalent to rate of carbohydrate oxidation, right? And destruction. Then plant cannot grow. So therefore, Rubisco is needed in large amounts for the plants to grow, okay, and survive. So it must have high rate of photosynthesis. So therefore, their protein is 50% is Rubisco. There are many other functions also Rubisco do. It contains ribosome and DNA molecules, right? These are the plastoglobuli. You see, these are plastoglobules. Sorry. These are black dots are plastoglobules, lipid bodies. You see, chloroplasts are not only synthesize carbohydrates, okay, starch. They also store out amino acid synthesis. They also synthesize fatty acids. You know, they are not only doing photosynthesis, okay. So chloroplast is actually the powerhouse for all kinds of synthesis, okay, for plant metabolism, okay, all right. The lipid synthesis. So chloroplast DNA also known as plastid DNA, circular double-stranded DNA molecule. Chloroplast genome is 120 to 270 kb, but usually 120 to 160 60 kb, one genome, one DNA, and has, of course, maximum genome size 217 kb. Contains about 100 genes to synthesize proteins. Only 100 genes. Even it could be less than that, around 100 genes. Okay. Do you think what mitochondria would synthesize? Mitochondria? You also a semi-autonomous organelle? Not it, or not? You also a semi-autonomous organelle. You also import protein from nuclear genome, right? Needed by chloroplast. It's we have plants are both chloroplast and mitochondria, but we have only mitochondria. Okay. We also have circular DNA in our body, right? Or not? In mitochondria, right? So you also have both prokaryotic and eukaryotic. Okay, as mammals. Okay. So all these exist. Okay. Chloroplast DNA includes large single copy and small single copy regions and inverted repeats. Okay. So mitochondria. Okay. Why going to mitochondria? Because mitochondria has genome size much smaller than chloroplast. It codes very few genes, which are mitochondria. It is much more dependent upon nuclear genome than the chloroplast. Okay. The chloroplast DNA region includes large single copy and small single copy regions. And inverted repeats, IRA and IRB. I'll talk about it. Conifers, a group of, they will lack inverted repeats. Remember that. Inverted repeats are not universal, but conifers and certain legumes lack this repeated inverted repeats. What is this? The LFC large the large single copy region. Are you able to see this? No, it doesn't. Okay. Large single copy region, LFC. It's so sensitive, black, this one, okay. Large single copy region, okay. And then a small single copy region, SSC, okay. And you can see inverted repeats. One, repeat here and repeat here. Okay. So called a repeat A and repeat B. Okay. Inverted repeat B, inverted repeat A. This is from tobacco, chloroplast. Okay. 155,539 base pairs. Okay, okay. Thank you. This will also respond. A little better picture of that. Okay. Think this is LSC, this is SSC, this one, and inverted repeats. Inverted repeats. A, inverted B. Okay. Yeah, this is a MATK is one of protein coding regions in multifunctional other than protein regions. MATK maps for two protein regions. MATK region is there also. We don't record it as a code. So general features, double-stranded circular molecule, right? No histones, right? It is prokaryote, no histones, but have bound proteins. There's some protein bound to it, but not histones. Organized into nucleides. GC content typically less than nuclear DNA. GC content is less than the nuclear DNA. Okay. Multiple copies, 30 to 100 per plastid, but some have more than that. So in one plastid, you have how many? 30 to 100 circular DNA. You understand that? So that is the challenge when you transform a chloroplast DNA, chloroplast transformation. You see, we are mostly doing nuclear transformation. Also, you are doing chloroplast transformation, but to a relatively smaller extent. Okay, and chloroplast transformation is one of the important features, and it is actually should be encouraged to a large extent now. With all our genetic concerns, okay, today about GMOs, genetically modified organisms, is that the pollen grain will escape. Let's say we have transformed a gene plant, okay, the pollen grain will go right of the transgenic plant, okay, and fall on the female of a gamete, stigma or style, whatever it is, and fertilization. And a non-transgenic plant will become a, suppose not become transgenic plant. Okay, that's the major concern. Okay, when the nuclear transformation, that is going to spread in nature. Okay, and then there are very often there are also cross species pollination sometimes. Okay, therefore is going to pollinate many other plants which are not the target. Okay, so this can target plant. So this can happen in nature. That's true. It can happen. There's no doubt about it. But if we do a chloroplast transformation, remember that same character you don't put in nucleus. We are not transforming nuclear gene, but a chloroplast gene. Okay, so the chloroplast is transformed, right? Chloroplast gene is modified, right? So whatever, let's say you are putting any gene into it, into chloroplast, it is going to synthesize inside the chloroplast. It's 70 ribosomes, right? Protein will synthesize. Right? Now that protein will give the function that is also can do the same work that is in the chloroplast cytoplasm. There are also limitations to that, but if we can achieve same function, but the chloroplast is maternally inherited, not paternally inherited, right? Chloroplast genome is not transferred. Chloroplast during fertilization, chloroplast is not transferred to the where? To the from the sperm to the or antherozoids or pollen grains, whatever you want to say, to the egg. Okay, it is excluded. Chloroplast DNA pair is not there. Okay. Only chloroplast not inherited. Only mother gives the chloroplast, mother plant. Okay. Same thing as mitochondria. Mother gives the mitochondria. Chloroplast, both chloroplast mitochondria are maternally inherited. Okay. The father plant doesn't give the chloroplast or mitochondria. The pollen grain, even though it goes kilometer, doesn't matter. It cannot give the what? Transgenic. It cannot fertilize, right? It will not have chloroplast to give to the mother cell. The pollination will be is there. Plant will not be transgenic, right? So this apprehension can go if we do the transfer of chloroplast. Okay. So therefore lot of energy also focus on chloroplast transformation. One of the reasons. Okay. Multiple copies. Okay. So if you think that is one of the multiple copies. Okay. 30 to 100 copies. Where we have two sets of chromosomes, right? Right? Two sets of chromosomes. I have a homeoplasm axis. So yes, only two genes, both have the same one gene, okay, two places maximum. But what are these? They can get 100 genes, 100 copies, right? So all 100 DNA, when they will be have expressed this gene. Okay, you have 100 times more protein, 50 times more proteins. Do you understand that? Because it is 100 copies of the gene. Okay, its own DNA. Each DNA will carry a transgene. The 100 genes are called the transgene that you are transforming. They are called transgene. Okay, these transgene. So there are 100 copies. The 100 copies is a proteins. Okay, so you have much more genetic material to support the protein synthesis. Okay, that to a genetically transforming. Okay, so it has much more expression. Of course, this is circular DNA of the chloroplasts. So some of the chloroplast genome coding genes, which is important. One is, of course, all of the RBCL, right? Rubisco small large subunit, right? You know that, right? Rubisco large subunit is coded by the what? Chloroplast genome. And a small subunit is coded by the nuclear genome, and post-translational imported into the chloroplast. So this is Rubisco is an example of a mosaic protein, right? One the large subunit coded by the chloroplast genome, and small subunit coded by the nuclear genome, post-translationally imported into the chloroplast stroma, where both LSU and SSU, or large subunit and small subunit, assembled to give the hollow protein called Rubisco. Okay, and this assembly also very interesting. You see, have you heard of a chaperones, a chaperone sample, molecular chaperones? Have you heard of them? Chaperones, no. There are many, these are proteins which are needed to give a proper conformation to the protein, can repair the protein and to give a proper conformation to the protein. They are not discovered in animals, they are discovered in Rubisco assembly in plants by John Ellis. What he did, so there is central dogma. What is that? That primary structure of the protein determine the secondary structure of the protein, right? Or not? Is it true or not? Primary structure of the protein determines the secondary structure of protein. That's the famous experiment on Finson, right? About Finson experiment. What he did, he heated RNAs, not it, to 95, 98, 99 degrees, lost its primary structure, secondary structure, right? And lost its activity also. Then he cooled it. RNA is a very tough enzyme, all of you know. They always destroy your RNA, right? When you do RNA isolation. So then what happens? They are say tough enzyme. So it also assembled back at cooling to RNA, and it could again destroy RNA. When heated at 100 degree, then these RNAs could not destroy RNA. Then it lost its activity, right? But when you cooled it back to 10 degree, 20 degree centigrade, 10 degree centigrade, then it got back its activity. So from this experiment, what? Anfinsen concluded that the primary structure and primary structure protein alone that determine the secondary structure of the protein. Not it. But this chaperones, molecular chaperones, qualifies it. Yes, primary structure is important, but not quite. Primary structure is important for the secondary structure of protein, but not quite. You need also some other protein, okay, for proper assembly of the protein. From this, so what he did the experiment for LSU and SSU assembled, and he found genes similar to GROW-EL and GROW-EF system of bacteria. Have you heard of molecular GROW-EL and GROW-ES in the bacterial system? They work as molecular chaperones. Okay, and chloroplasts have only ancestry of the prokaryotic ancestry. Right? So these they inherited from bacteria, cyanobacteria. Right? Cyanobacteria. So the molecular chaperones, the chloroplast inherited from cyanobacteria, okay, exist today, okay, and they are responsible for assembling large subunit. What is Rubisco? 8 large, 8 small, right? In eukaryotic system today in higher plants. So 8 large subunits and 8 small subunits assembling to form a visco. And this assembly is not taking place automatically. The assembly is mediated by molecular chaperones similar to GROW-EL and GROW-ES present in bacteria. Okay. A multimery complex. Okay. All right. So this RBCL is synthesized in the where? In the where? In the where? Chloroplast genome. Right? Right. What is RBCL? Rubisco large subunit or RBCL gene. Okay. ATPB, beta subunit ATP synthase. What is ATP synthase? Komal, I am on leave for 3 days. Okay. So, so ATPB, large single copy region chloroplast, okay, is beta subunit of ATP synthase. We function ATP synthesis hypothesis. You know that, right? ATP is complex, what you're knowing by now, right? If not, okay, you'll know in near future somewhere, okay. So this also coded by the chloroplast genome. So there are also other ATP synthesizing genes that are coded by chloroplast. Only that. These are only simple few examples. MAT kinase, large single copy region of the chloroplast. What it does, it maturates, all responsible for the maturation of the genes and all that protein. Then you have NDHF, single copy region of chloroplast. It is a small one, subunit of chloroplast NDH dehydrogenase, which functions in the converting NADH and DH, various reactions and respiration. Sorry, Gold Tower. So this all talk about little bit, not what endosome binding hypothesis, how, you know, this is a eubacteria, this is a eukaryote, anaerobic eukaryote, right? You see, eukaryotes are anaerobic is did you know that? When chloroplast evolved, before chloroplast evolved, life was full of anaerobics. We'll talk about later, one class. We'll not talk about that today. Oxygen atmosphere, effort for oxygen, cell enlargement. Then you know proto-eukaryote. This has a nucleus now. Okay. This evolved around 150 million billions of years ago. Sorry, 1.5 billions of years ago. Then became a cyanobacteria, a purple bacteria invaded. Then it became a mitochondria. Okay. And then cyanobacteria invaded it. Then become a chloroplast. Okay. You understand that? The mitochondria have prokaryotic origin. That means this has happened because of the invasion of a purple bacteria to the eukaryote system, anaerobic eukaryote. This is anaerobic eukaryote because you could not respire, right? There is anaerobic eukaryote, right? Who survived, could not respire, right? Following absence of oxygen. Okay. Maybe rudimentary respiration was taking place there. Otherwise, but then when mitochondria invaded, when the purple bacteria invaded, then it became more and more oxygenic, okay, and respiration started. Then came invasion of chloroplast, of cyanobacteria, they became whole cell, then it became single cell, like single cell organelle like aminobutyric. The Campinomonas evolved to multicellular what? Algae, and so on and so forth. In the process of evolution. They're all determined by oxygen. We'll talk about that later. These are three famous, I would say, in chloroplast biologist, plant molecular biologist, chloroplast plant molecular biologist, Ruth Seger, Lawrence Bogorod, and Rochex from Geneva. Okay, they are contributed a lot to molecular biology of chloroplasts. So either chloroplast double membrane envelope, stroma, thylakoid membranes, interthylakoid space, and lumen, right? How many compartments it has? Outer membrane, right? Intermembrane, then between outer membrane and the intermembrane space, 3 compartment, 4th compartment is stroma, 5th is thylakoid membrane, 6th is thylakoid lumen, right? Okay. So this is what a chloroplast looks like, of course, charge granules and all that. And I'm sure you know the freeze factor studies, for localization of photosystem 1 and photosystem 2, the thylakoid membrane, right? That came with freeze factor studies showing differential distribution of pH1 and pH2 in the stacked and unstacked region, right? You know about this? You know or not? Photosystem 1, photosystem 2, they are not uniformly distributed in the thylakoid membrane. pH1 in the unstacked region and pH2 in the stacked region. Okay, all right. And there is region to it. So do you know the reason why? Do you know reason why pH2 is in the stacked region and pH in unstacked region? Tell me. Less wavelength. Less wavelength. Wavelength will not distribute the organelle differentially. pH has more negative charges and pH2 has less negative charges. And therefore, when there is magnesium in the chloroplast, when salt in the chloroplast, they seal the negative charges better and it's kind of stacking. I will go to that. Okay. So first to understand why granite stacking takes place. Okay. All right. Why the stacking of granite anyway? So thylakoid membrane from granum. Why? What is the mechanism? How do they unstack? It's dynamic nature. See, all membrane dynamic. It is a bilayer lipid membrane, right? Let's say this is the bilayer lipid membrane, right? Yeah, bilayer lipid membrane. This side, 2 layers. But the lipids are also here, right? They are in constant motion, right? All the proteins in constant motion. PS1, PS2, LSCP, all those cytochrome BF complex, they are also moving. ATP synthesis, they are all moving. They are nothing dynamic, nothing static, right? They are all moving in the membrane plane, right? Thylakoid membrane. And that's the dynamic nature of stacking and unstacking of thylakoid granum to become thylakoid or stacked from thylakoid. Granum is important. Yeah. So what do they do? Photosynthesis, starch synthesis, I told you, fatty acid synthesis, right? That you say plastoglobules, right? Amino acid synthesis, pigment synthesis, chlorophyll carotene synthesis, nucleotide synthesis, nucleic acid and protein synthesis, sulfur nitrogen assimilation. Think about it. They are not doing almost all the things needed for the plant. Okay. Not only photosynthesis. Okay. Its own genetic system, of course. And you see how the chlorophyll A and chlorophyll synthesis takes place. The chlorophyll synthesis takes place only during light time and goes and is not synthesized during dark. Okay. This is in night time, this is dark time, right? This is taking place in day and not taking place in night. Why do you think so? Why do you think so? Why chloroplast synthesis is not taking place in night? Only in day. Why? If you grow a plants in dark, it will be green, it will be yellow, right? Right? It is not green, right? Once you transfer it to light, it will be green. Not it. And right. So etiolated plant, okay, will convert to become a green plant. Right? Why? What determines it? There are many factors determine it, but one important is chlorophyll biosynthesis itself. There is a gene called POR, protochlorophyll oxidoreductase. Do you know about anything about chlorophyll biosynthesis? What is the substrate of chlorophyll biosynthesis? Glutamic acid in plants. In animals, the glycine succinyl CoA. Animal. What ALA does, delta amine ribulinic synthesized. What do they produce? Glutamic acid synthesized ALA, delta amino ribulinic acid. ALA is metabolized to protoporphyrin 9, right? If magnesium gets inserted, series of steps. Then magnesium gets inserted, becomes chlorophyll. Where iron gets inserted, becomes cytochrome, right? That protoporphyrin 9, iron is center, becomes cytochrome. Protoporphyrin 9, magnesium at its center, become chlorophyll. Now, protoporphyrin, once you become magnesium protoporphyrin 9, so it plants both magnesium path, magnesium branch, and iron branch, both are functional. In plants, in animals, magnesium branch is shut down. Only iron branch is functional. So once magnesium inside protoporphyrin 9, magnesium protoporphyrin 9, they become MP, then become protochlorophylite. Protochlorophylite has converted to chlorophyllite, then chlorophyll. And it needs light for the photoconversion of protochlorophyllite to chlorophyll. It needs light. Okay. This is light dependent enzyme. Okay. This enzyme can function only in presence of light. The enzyme called POR, protochlorophyll oxidoreductase. Only function during light. Therefore, you see a lot of chlorophyll synthesis during light time. If you stop the light, there is no synthesis of this enzyme becomes inactive because there is no light. Absolutely dependent on light for its activity. The protochlorophyll will not be converted to chlorophyllite or chlorophyll, and chlorophyll biosynthesis stops. Okay. We said this we have seen all nuclear division. It's a chloroplast division. Okay. A chloroplast dividing 1 to 2. You see that? First there is back here, then they are all separated into. Inverse proteins FTS, immersion mediated by division bacteria. Remember, it's division mediated by FTS, a division led by bacteria. That's why I'm showing this slide to you. Okay. Because of prokaryotic origin. For chloroplast division, you need more FTS-like protein, and they are present in the chloroplast gene. Okay, all right. And this FTS, if people have tried to modulate FTS content in plant, okay, in chloroplast plant, you can also synthesize it. The FTS, if you increase or decrease, you also see that number of chloroplasts will go down if FTS is decreased, and if it will go up, FTS is increased. The FTS-like proteins are involved in chloroplast division. So if you increase by transgenic approach, if you can increase the FTS content in plant, what happens? divide more and more chloroplast, divide more and more, the number of chloroplasts is going to increase, right? But if you slow it down by antisense approach, okay, or some other approach, RNAi approach or whatever, or PTGS approach, then you will see now it's slowed down. Number of chloroplasts will be less. We can modulate the number of chloroplasts per plastid in a per cell, okay, by modulating also FTS gene. Okay, do you think if we increase the FTS, plastid content or chloroplast content in plants, okay, what is going to happen? Will you photosynthesize more? Huh? We increase the photosynthesis chloroplast content in the plant, right? Increase the overexpressed FTS, right? The protein content increased, right? Protein content increased, and what happens? We have more chloroplasts, right? The more number of chloroplasts, what is going to happen? Think about it. What's in the electron transport will be more? Not it. But what will be less is Rubisco. You more light reaction, but protein content of the plant, other protein remains same. Rubisco is not going to increase. Okay, you are plant synthesizing 50% of its proteins in one protein called Rubisco. How much protein it can increase? Cannot make it 100%, right? So you cannot synthesize as much Rubisco as possible. Okay, therefore they may have more number of chloroplasts, but Rubisco will be limiting. Okay. Therefore, the seed fixation will never increase. Otherwise, you could have increased rate of photosynthesis by increasing the number of chloroplasts. There are several forms of plastids, proplastids, because of plastids found in meristems. The etioplasts that grows in dagron plants, right? Then a chloroplast, green tissues, the amyloplast prominent in roots and starch, and chromoplast. Okay. Different kind of plastids present in plants, right? Any plant you take. Okay. When the plant germinates under the ground, right? Germinates under the ground, na? Then it comes up, right? In most epigere form of germination, right? So when it comes up, under the after germination underground, not getting in light, right? So its development there is light independent. Okay, so light independent development taking place under the soil or in dark, right? And this is undergoing scotomorphogenesis. It's called development in dark is called scotomorphogenesis. So it will have a long hypocotyl, right? Become tall. Why it becomes tall? So that it comes up to the ground. Push is that it comes penetrates the earth surface and comes to the top. Therefore, all diagonal plants become tall, even if they are not growing under soil. Okay, even if they are not growing under soil, just grow them on the PTDs and put in dark. Okay, they will become tall. Terminate in PTDs, that they will become all tall. Why? Because the impetus is given in nature to these plants, so that they can grow fast, become taller in the. Why does it grow tall? Because seed has very definite amount of endosperm. There are not infinite amount of endosperm, very small amount of endosperm that they can eat during germination. But then they have to synthesize their own food. Okay, Only when they come from top, from soil to the top of the soil, then they cotyledons will get light, right? Can develop the chloroplast, okay? They can photosynthesize. So therefore, dark grown plants are usually taller than the light grown plants. Okay. So therefore, they are called proplastids because all plastids found in meristems. You know meristems, meristematic tissue, but cell division taking place. You have cambium also, right? But division takes place. And if you say dicot and monocot, they are different localization. A leaf, let's say monocot leaf, barley, or rice, or wheat. Okay. So young cells are in the bottom of the leaf, and old cells on top of the leaf. You know that because the meristematic cells are present in the bottom. Okay. They start dividing and form number of what? The leaf grows and grows and grows. So young cells are in the top, right? Young cells are in the bottom, old cells in the top. Okay. So if you see the gradient there, you take the take a section from the bottom of the leaf, again 1 centimeter after that, 1 centimeter after that, 1 centimeter after that. You will see different developmental stages of the plastic. Okay, not single leaf. That is the beauty of it. A monocot leaf. They are proplastids. Ethioplasts, I told you, the plastids that grow in from isolated from the dark grown shielding called ethioplasts. Okay, ethioplasts. They are chloroplasts, but ethioplasts. So then there are chloroplasts. All of you know that, all green tea. Then amyloplasts in the roots. They store starch and colorless, right? With their colorless. Chromoplasts, you see tomato, right? Green in color, right? Tomato green in color, then becomes red. Okay, okay, during so-called fruit senescence by loss of chlorophyll and appearance of many xanthophylls, carotenoids. All right, the chromoplast. In mature fruits, not carotenoids, little chlorophyll, right? This is telling about, you see, right? Youngest in the bottom, oldest at the top, right? You can see how the plastids, if you look at the plastid here, it will look like this. Plastid a little above will look like this, and this. Look at this. Okay. How the plant development, plastid development, taking place in a single leaf. Okay. You can see the gradient of development pattern of the what? Of the plastid. Okay. At different parts of the plant, parts of the leaf. So this is proplastid, the mature ethioplast. Why still ethioplast? It's not in the light yet. All happening under the below the soil. Under the soil. Okay. Scotomorphogenesis. Then it has come above the light, right? And it becomes chloroplast. Okay. You can see these are called prolamellar bodies. Prolamellar bodies. And they're dispersed into a lot of thylakoids and all that later on. And this is amyloplast. You see amyloplast roots I told you. Have you can you see can you tell me a root who stores starch? Root. Potato is not a root. Potato is a stem. Tell me root. Huh? Huh? Tell me a root. Asparagus? Have you seen asparagus roots? Huh? Asparagus roots. Huh? There are many roots like that. Carrots. Not it? Huh? Beetroot. What I am showing you, you can see rudimentary thylakoids also here. In the where? In the amyloplast. See that? Rudimentary thylakoids here. In the amyloplast. Okay. So this is, and this is chromoplast. You can see how the color changes, right? Green to red. Okay. Or this is a green fruit. This is a yellow fruit or red fruit. Okay. The young chromoplast developing into it. Okay. Still green, but developing into a chromoplast. Okay. So lot of plastoglobules, thylakoids disorganizing and forming plastoglobules. Okay. Lycopene crystals and all that formation. So proplastids, phytoplasts, right? Right? Then light, chloroplast. The proplast can put out to chromoplast. A chloroplast can also go to chromoplast. Okay. Roots, proplast to amyloplast. Okay. The development, right? The development that takes place. Okay. Inheritance is uniparental, as I told you. Only maternal inheritance, right? Okay. Father does not contribute to chloroplast. In chlamydomonas 7, where the two cells fuse, chlamydomonas, is not that kind of development that higher plants have. Okay. Two vegetative cells, having little bit sex determination there, plus and minus strain we say. Okay. Okay. There, when they fuse, still chloroplast, paternal chloroplast is destroyed, DNA is destroyed. Okay, during fusion. Paternal. So you see the inheritance that has taken right from chlamydomonas when they are unicellular organelles but fusing together. We talk about now, okay, after millions of years evolution. Okay, right? But origin you see, no, many origins may have many things before that also. Okay. But I give an example, chlamydomonas unicellular organism. Okay. When they fuse, the paternal or father chloroplast DNA is destroyed. Okay. Maternal chloroplast DNA preferred and replicated. In some land plants, the paternal plastids are excluded during fertilization or absent from the small cells. Essentially, all plastids are DNA, usually same DNA throughout the organism, homoplasmy. It's important, homoplasmy. Homoplasmy is important, okay, for chloroplast DNA, otherwise it's very variegated, okay. Okay. If some chloroplasts, you see many are, variegated, different colors, Some chloroplasts are okay, green, some chloroplasts lost their chlorophyll. Okay. So all the chlorophyll present in the chloroplast only. Break. Okay. Hello? sir. Yeah. ठीक है, ठीक है. Hey, what was this? Doesn't matter. I'll find out. Yeah, here, here. That's simple, simplest. You go, you go, you go. So talk about, how the paternal DNA even get destroyed. Okay, during even in chlamydomonas. Okay, so the same trend goes on forever. Okay. All right. So as I told you, essentially all plastic DNA, okay, usually same DNA throughout the organism, right? Same, okay, copy of the other. Right? That means copy of the other, okay? Same genetic makeup, okay? The DNA sequence does not change during differentiation. Chloroplast DNA sequence also does not change. And no DNA sequence changes in our body also, right? We can have a chloroplast differentiation, right? Or etioplast, chloroplast, or chromoplast, okay? So if you haven't seen chloroplast in a chlamydomonas, the zygote, but here also, you see, as I told you, paternal is destroyed. I can show you one mitochondrial, maternal chloroplast DNA destroyed here from zygote, okay? All right, inside zygote. I can show you evidence here. The zygote, right? The zygote has one paternal and maternal, right? Component paternal DNA, maternal DNA, right? Now you can see the yellow dots are eh, is a big problem. So these ones, are getting destroyed. We'll see the DNA. This is existing from maternal DNA. Maternal DNA, you'll see now all these arrows, they are no more here. They are gone. Okay. The fluorescence labeling, we can see that DNA is gone. The fluorescence labeling. So giving a direct experiment, saying chlamydomonas also, maternal, paternal DNA is getting destroyed. Which DNA? Chloroplast DNA, not the mitochondria, not genomic DNA, right? The chloroplast DNA getting destroyed. And this is attacking acetobularia, okay. One nucleus at the bottom. This is 5 centimeter long, single giant cell. One cell 5 centimeters. Think about that. Of course, there are many pinocytic alga like that. Okay. but but that's not important. What is important here is that it's regulating, huge, how many? At least 1 million chloroplasts in a single cell. Okay, and one nucleus is regulating 1 million chloroplasts. That means think about its transcription ability, gene expression ability. Okay, but it has supply the proteins to the 1 million plastids, right? Rubisco some small subunit to 1 million plastids, ATP synthase, many subunit synthase, and many proteins, many proteins, right? LSU and all, SSU and all that. I told you 95% proteins are coded by the nuclear genome. So 95% protein is supplied to the chloroplast. But so one nucleus has to have so much gene expression capability, okay, the messenger RNA produced, and its synthesized and translation ability in cytoplasm to support so many chloroplast development, chloroplast function. There are so many photosynthetic organisms in fungi and animals. Can you guess an animal which is having chloroplast, chlorophylls, animals? Huh? Corals, right? Flatworms, sea sloths, salamanders. Okay, animals. Fungi, lichen is fungi is lichen, right? It has kind of bacteria, algae in it, right? This is lichen, right? Fungi and algae together, right? These corals, right? Green in color here. Good, it has chloroplast. Okay. Right? Another worm here. They are also chloroplasts. Can photosynthesize own. Salamanders, you see, it is vertebrate. Salamander amphibian. Yeah, vertebrates. And these vertebrates are also by chloroplast body. You can see these yellow spots, so-called yellow spots. Many egg capsules. So first important. What is this? So this is again endosymbiosis. Okay. Some algae have come and invaded the salamander, living inside the salamander. Okay, symbiosis, right? Because spotted salamander, huh? Sea sludge. Okay, chloroplastic sea sludge. You can see the chlorophyll fluorescence, red chlorophyll fluorescence. Sea sludge, the chlorophyll. Okay. Okay. So a plant grows in dark. It undergoes photomorphogenesis, right? And when it goes to light, it undergoes photomorphogenesis, right? Is it understandable to you? Okay. So there are many photosynthetic pigments that are written down, which also mediate photomorphogenesis. Phytochromes, cryptochromes, okay. And these are two main photoreceptors who are responsible for photomorphogenesis. And also movement and all that other phototropins are movement, right? So one in dark development, one in light development. Plant grows in dark, called what? Etiolated, right, plants. And as I told you, yeah. What is the dark grown plants? I told you long hypocotyl. This cotyledon, below cotyledon is called hypocotyledon, right? This cotyledon, below this cotyledon called hypocotyledon. See, big hypocotyl. Look at a light grown plant have a small hypocotyl. Okay? Alright. I told you reason why a dark grown plant has long hypocotyl. Right? Right? Because it stems from its original process of plant growth under soil. Right? It must grow very fast to bring the cotyledons to the light in the surface of the soil, so that they can get light, undergo so etioplasts will become chloroplasts, and it becomes chloroplast. It can photosynthesize and develop. Okay, plant can growth can happen. Okay, these are cotyledons, right? So what happens when it is etiolated plant? So like a de-etiolation, forming a green plant, right? Process. So these cryptochromes also can do the same function, come flowering, stomatal opening, shade avoidance response. Okay, all this come there, and it will be blue light, and this is all about cryptochromes. I think, have you heard of cryptochromes? Have you heard of them or not? What are they? Huh? Yeah. What do they contain? Hmm? The flavins, flavin adenine dinucleotide. Flavin. The protein have binding to flavins. These flavins can absorb in blue. Okay. Phytochromes absorb in red. Okay. Cryptochrome absorb in blue. Okay. Okay, I'm giving an example here. This is about how our chloroplast development taking. We are talking how chloroplast development modulated by photoreceptor and light. Okay, so for modulation of light plant growth development and modulation of chloroplast development, you need what? Light, and light are photoreceptors, and the plants have many photoreceptors, phytochromes and cryptochromes. One to the phototropins that is needed for plant movement. Okay, when plant grows from light bends towards light because of the phototropins. Okay, here is showing you a wild type plant grown in dark. Okay, a cryptochrome mutant grown in dark. Okay, then cryptochrome plus plant have again phytochrome B minus plant. Phytochrome B is also absent here. They are almost similar, slightly different, almost similar. Now we are giving them blue light or red light. What happened to blue? You can see if you give blue, we will be going in blue light, wild type plant going normal, right? Cryptochrome A mutant -1 mutant, it doesn't have much cryptochrome. Okay, all right. So you can see plant is growing but still mimicking long hypocotyl. You see that? Mimicking, except that it's green in color. The green in color of the leaves are green in color because you got light. Okay. So because there's a many colors of light, and the light regulates for chloroplast development, and chlorophyllosynthesis can take place in light. Even it can take place in blue light, no problem. So it is green in color. But it still have long hypocotyl. Okay, this is the photomorphogenesis. So okay, it's not able to perceive light, blue light. Okay, this is Cry1 plus mutant. Cry1 plus mutant means where you overexpress Cry1. Cryptochrome is overexpressed. Okay, you can see it's shorter than the wild type. Okay, cryptochrome is overexpressed. And it's a 5B mutant, phytochrome B mutant. We are growing in blue light. We don't care whether phytochrome is there or not. Okay, because there is cryptochrome there. So cryptochrome can do the same function as that of phytochrome. Okay, so it tells you as regards chloroplast building development is concerned, 5B and cryptochrome will doing the same similar in right. 5B mutant, you can 5B mutant similar to wild type. Okay, but now same plants are growing in red. Okay, same plant. You grew them blue, now give them the red. The wild type plant, the cryptochrome mutant, it is cannot see what blue light. Okay, but there is phytochrome. Okay, you can see red light. So it is developing normally now. 5B mutant, you see the cryptochrome mutant, normal development. Because it may not see blue light, may not have blue light, but have phytochrome. Or you get a red light. Now cryptochrome 1 plus, no problem, same thing. Cryptochrome overexpressed, but in red light, it doesn't matter. But if it's 5B mutant, still having this long hypocotyl. Okay, this is a 5B mutant, tall, right? 5B mutant tall, because we are growing in the red light. Okay, and 5B mutant is what? Doesn't have phytochrome, is not able to see red light. So it becomes tall, behaving like a tall plant, like a dark grown plant. Huh? You can? Yeah. A shade plant is similar. Shade plant is different. A shade that that is phytochrome there. You want a mutant here. Phytochrome still there, but they have different response. You see what happens if you go to forest. Have you gone to forest? Any of you? Or shade habitat? Or you go to any in the campus, a lot of trees, right? So there are tiers of plant, tall plant, there is small plant, still a small plant. The top plant is getting all the lights, and the plant going in the bottom getting less light. So the different light intensity, right? In nature, look in nature, okay? In less intensity. Not only that, the top leaves are harvesting all the colors of light from what? Blue to red, violet, indigo, blue, green, yellow, orange, red, right? All lights that they are able to observe. What? Except green. Why not green? Because chlorophylls are green. It cannot absorb any green light. So all the green light will come down. Let it. If all the plant grows in top, okay, observe all the other colors light, not green light. So most of the green light will come to the bottom. Not it. All the red light they will absorb. What will come? The far red light to come to the bottom. Okay. So plant growing at the bottom is deficient in red. Red light absorbed by the top leaves. Not it. Deficient in but deficient and rich in far red. Rich in green. Not it. Right. So the far red light will be so I will for the present neglecting green here. But the red and far red counter phytochrome. Okay. That is shade response. It is a question to your. This is my answer to your question, right? Listen, I'm answering to your question only. So you have a red and far red now changing. What is red? What red light? The phytochrome absorbs red light. Put PFR, right? PFR comes back to PR. Okay. So PFR by P total that governs plant development. Not it. So if a plant is getting more far red, plant going in the bottom by more far red and less red, guess what will happen? What FR does? Convert PFR to PR. PFR is PR, isn't it? So the ratio of PFR by total phytochrome is going to change. PFR is going to be less or not? Or not? Do you have no phytochrome or not? Right? Absorbs phytochrome absorbs red light becomes PFR form of phytochrome. PFR form of phytochrome absorbs far red light and comes back to PR form. Right? Right? PFR is generally considered active form. Always. So what happens when PFR accumulates? So PR in far red enriched light, shade habitat, a lot of far red light. FR will convert back to PR. Therefore, PFR or P total ratio is going to change. That is encourage shade elongation, stem elongation. Okay. So yes, shade habitat also encourages what? No, plants be longer. Why they have to longer? Because they have to go to the sun. Okay, to get the light. Not that plant want to become taller, because plants have the ability to become taller because of the change of red and far ratio that happening in the bottom. Okay, but once you let's say they become tall. Okay, now you cut the trees. What is going to happen? They will go to all the plants. They're getting taller, but tall plant cannot become small again. Remain as tall. Okay, when you cut the forest, when you part the forest, make it. Okay, there's no reversal of it, right? Yeah.
Plant stratification, like height in the stratification in the height. So the lower height plants are getting far red one.
More far red.
More far red, yeah. So is it like kind of disturbing the proportion of PS2 and PS1? And in
PS to
biogenesis
PS1
case,
ratio is different here. Yes, that will have another kind of adaptation of chloroplast
yes,
itself.
yes, sir,
Okay. So therefore, if plant grows in low light, will have accumulate more chlorophyll. Why? Why do you think plants have grown low light have more chlorophyll? Because in the more light. Okay. So it has harvest more sunlight and transfer to reaction center to photosynthesize. The plant is grown low light or shade habitat, it should have more chlorophyll. In turn, it should have more light harvesting complex. Light harvesting complex bind to chlorophylls. Not it. So LSC will be bigger in plants which are growing in shade. Right. Therefore, A by B ratio is going to change because LSC is rich in chlorophyll B. A by B. If you see a plant extract chlorophyll A by B ratio is how much? 2.83 sometimes. But if you take out LSC, light harvesting chlorophyll complex to B, LSC2, it's chlorophyll A by ratio is 1. Okay. You got 2.8 to 1, right? 1. So that means more relatively, LSC has more chlorophyll B. Right? So more light harvesting chlorophyll content, right? More chlorophyll B it will have, plant will have, because it harnesses all of solar energy. What chlorophyll B does? Only harness the light and transfer to chlorophyll A. Right? Transfer to chlorophyll A. Right? So chlorophyll B increases to harness the solar energy, okay, and transfer it to chlorophyll A. So chlorophyll B content increases. Okay. We have to increase antenna size Antenna you know right by now. Huh? So in antenna side, so light is observed and transferred to the reaction center. Has anybody talked about holistic photosynthesis? Nobody? From top to A to Z, an introduction. Huh? Light reaction summary. It's very important. Okay. All right. It doesn't matter. Okay. All right. So you can say light regulation of plant development and chloroplast function. And this also goes to same goes chloroplast development. Okay. A color blind mutant arabidopsis. Can color blind mutant? You call it color blind. Blind to these are color blind, right? If they have a mutant, they are color blind, right? Some are blind to blue color, some are blind to red color, right? They are color blind. Okay, they cannot observe blue light, see blue light or red light. Yeah, anthocyanin also cryptochrome. You know have more anthocyanins. Okay, cryptochromes also regulate anthocyanin. What are anthocyanin? What do they do? Huh? What is their function? Protect the chlorophyll. How? How? Yeah. Anthocyanin part gives the color to the leaves, flowers. देखो red flower, blue flower. देखो ना, they are different anthocyanin. Okay, more anthocyanin means more color plant, right? So they determine the color of the flowers, color of the leaves. Okay, all right. So and nature, anthocyanins are developed in nature, really, not to protect plants, but to attract insects for survival, for germination. Okay, for pollination. No pollination, no race diet, right? You don't do. You go to a forest, or there's some. You go cut some trees. Okay, you don't cut a tree. You also caught thousands of insects living on the tree, right? And what these insects do? Go and pollinate the flowers. Okay, you cut the one tree or few trees, you destroying so many insects, their habitat. So that will reduce your pollination rate. And if the certain particular, and if the plant is responsible, dedicated for pollination certain insects, only certain insects, then those insect dies, means plant dies. Plant becomes extinct. There is no seed formation, right? Plant becomes extinct. So so-called, many plants who are endemic plants, who are located in one region. Okay, they will not find in other parts of the globe. The much chance of those plants becoming extinct because of climate change. Okay, because you destroy the habitat, not on climate, if you man-made, not human interactions in nature, not climate change. Okay, you go and destroy the habitat, plants are gone. You will not see them anymore. Do you know only since origin of Earth, how there are millions and millions of species who have evolved, right? How many species have survived? How many percent of species have survived today since the origin of life? 1%. 99% of species have become extinct. Okay, they evolved and become extinct and gone from the earth. Okay, So that's the role that is not man-made in climate change. Man is evolved only 1 million years ago. Okay, what we are destroying the climate, changing the climate, you can see about only a few last 1 million years ago. Earth history is billions of years. Earth originated 4.5 billion years ago, 4.6 billion of years ago. Okay, so still climate is changing. Many regions. Okay, this climate change, extinction events, geological events have destroyed, many life forms. Of course, we are multi-compounded as human species, Homo sapiens species, have destroyed so much climatic conditions, changed the climatic conditions, destroyed plants, destroyed trees, made, destroyed forests. These are one of major reasons why climate change is so prevalent now. What if you go to forest? What do you see? Have you seen the temperature in your campus and outside campus? It's cooler here. Right? Why? There's so many trees around. The trees are transpiring. Right? So they are actually natural air conditioners for you. Okay? They are transpiring. They are, the latent heat of vaporization provided by the leaf. Okay? The working vapor, cooling the atmosphere. Okay? Therefore, you are cooler here. Once you go to outside, there are no trees, they are gone, so temperature has moved higher. You same thing go to GIAN campus, full of trees, you go to campus, at least 5 to 6 degrees below the ambient outside campus. Okay? That happens because the trees there. So so-called temperature increase, high temperature increase. One of the major reasons is due to destruction of trees, destruction of the forest. Okay. Not only that, forest, what do? Absorb carbon dioxide, the process photosynthesis convert them to carbohydrates, and they store for millions of hundreds of years. Many trees live for 100 years. Can you give an example of high long living tree? Banyan tree how long lives? How much? Bristlecone pine. Bristlecone pine. It survives 5,000 years. 5,000 years. It's still there in California forest. 5,000 years. Right from Indus Valley civilization standing. Okay? The time of Indus Valley civilization. Okay? So this is, I won't talk about phototropism here, for negative and all that. I skip this slide for you. I'm not talking about phototropism, but I'm coming. Again, phototropism, I'm not going to go that. And to come to our phytochrome, who discovered phytochrome from this response, right? Remember this experiment? Let us terminate the experiment. By whom? Who did this work? Sorry. Here. Anyway, don't bother about that. Who discovered phytochrome? Who discovered phytochrome? Hendrick. Remember Hendrick? I don't know. Here it is. These two guys are Borthwick and Hendricks. Hendricks and Borthwick. Okay. They did this experiment, famous experiment of lattice seed germination, red and infrared light, and found discovered phytochrome. Okay. Then later on phytochrome. Now we know phytochrome is only one form. There are many forms, many coded by different genes. Phytochrome A, B, C, D, and E genes, right? All we know by now. Okay. These are phytochromes. Tryptophan, flavin, billions. Okay. Okay. So this is, as we were talking about before, PR form phytochrome, PFR form of phytochrome, right? So that's questions, right? So PFR form, if PFR form observes the blue or far red light, it becomes PR form. PR of the red light becomes PFR form. So PFR by P total govern the plant development, okay, and photomorphogenesis. Okay. Yeah. So do you know where phytochrome is synthesized? Phytochrome is synthesized, one part synthesized in the plastic, okay, the chromophore, okay, and the protein part synthesized in the cytoplasm, okay, then assembles. Then there is assembly. Okay. But the chromophore phytochrome is the open chain tetrapyrrole. Open chain tetrapyrrole. Chlorophyll is a closed chain tetrapyrrole, right? Like this, right? Tetrapyrrole, 4 spiral rings. Closed chain tetrapyrrole. Whereas phytochrome is an open chain tetrapyrrole. Okay. Cut from the top, it will open, right? Not chlorophyll, but protoporphyrin 9 maybe. Cut from the top, you pour tetrapyrrole ring. Okay. So phytochrome of this is phytochrome synthesized from the chloroplast, phytochromobulin, then it goes out from the chloroplast to the cytoplasm, where this gene, phytochrome gene is coded by the chloroplast gene, mRNA comes out to the nuclear pores in the cytoplasm and translated, and then it assembles into the phytochrome in the cytoplasm. Okay. I don't want to go to chromophores and all that. That's not, not my topic now. Just migrate to that topic. Where is my here? And I just, with the pen drive, I kept it back. I changed my pen drive. Okay. To a different topic now. Because two different lectures, I put in two different. Okay, now let us start. Okay, so we are talking about a few minutes ago about climate change and biodiversity, right? Climate changing, biodiversity decreasing, and so on and so forth, right? Okay, yes, it has true. Let us what a trigger for biodiversity. The greenhouse earth that triggered biodiversity. But a greenhouse and ice house, greenhouse where temperature is high, right? And ice house where temperature is low. You see, greenhouse earth, when temperature is higher, diversity is higher. Biodiversity is higher. Not beyond certain region, but within the limit. If you see, go to in the ocean, if you go to the tropical ocean, where in tropics, okay, in Africa and between Asia and all that, okay, tropics. Number of species present, marine species present per square kilometer or square volume of water, or cubic kilometer of water, okay, is more in tropics and less in temperate zone. Why? The high temperature always has promoted biodiversity. Okay. So therefore, biodiversity in Africa and India is more, and this is one of the reasons why probably all human beings evolved in Africa. The rate of mutation, rate of evolution is faster in tropics. Okay. Therefore, if you look at our origin from Africa, from starting from Homo erectus to Homo sapiens, okay, all took place in Africa. Okay.
So this higher temperature, how it impacts when we consider it in terms of the plants? Plants are algal photosynthesis.
Yeah, same thing plants.
Because when we see
Same plants. Same thing of plants also. Evolution में same thing of plants also. The biodiversity in tropics is much more than biodiversity in temperature. Then comes oxygen metabolism and photosynthesis. Okay. It has really promoted our biodiversity. Actually, the forbinger of our evolution, photosynthesis, and plant evolution. And our talk about that. Competition selection forces evolution, of course, okay, and evolution. Increase and decrease of niche, right? Decrease of niche means what? Feeding region, I can grow, okay. Decrease habitat, okay. Decrease of habitat will lead to extinction of species. Increase of habitat will go to proliferation of species, right? And of course, extinction events that has largely affected our biodiversity. Lot of species become extinct. As I told you right now, 99% species become extinct. If all the species would have survived, okay, right from origin of Earth to today, the biodiversity would have been much, 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 much higher. Okay, all right. The extinction events have really determined the biodiversity. Okay. You can see your example. Okay. This is number of familiar species, and you can see from here to here, we are going back only to 500 millions of years ago. Okay. 500 million to today. Okay. Okay. As you can see, number of species increased. Okay. And these are all huge extinction events. Sudden drop in here you see decrease, again decrease, again huge decrease here, right? That due to extinction event. You see, large was why geology has determined life form, geological events, with earthquake or any igneous explosion, okay, or volcanoes, volcanic eruptions, okay, that have determined extinction events. Okay, earthquakes, huge earthquakes. Okay, we're not talking this earthquake, huge earthquake. So now Earth is a little cooler, temperature-wise, than what it was origin of Earth. Okay, unlike so you can see, therefore, for then you can see biodiversity is high today compared to our geological past. So what do you understand? Current climatic situation has favored our biodiversity. Okay, current climatic conditions, right? So current climatic conditions have favored our biodiversity. Biodiversity high today. What happened in the origin of Earth? 4.5 billion of years ago, full of carbon dioxide, okay, full of methane, and no oxygen, right? No oxygen. That's what the life. That is the Earth today. In the Earth in the early years, right? There is no water also. Water came to Earth by many asteroids collided with Earth and brought water to Earth. Okay, then Earth. The sea and all you see are not there. It happened after collision of asteroids with early Earth. And what is there? Full of carbon dioxide, methane, oxygen. Around 3.5 millions of years ago, 3.5 billions of years ago, came cyanobacteria-like organism. Cyanobacteria-like organism. Okay. Who saw them strobules inside rocks. Inside rocks, we saw the strobules. Okay. Cut the rocks, you find those fossils of cyanobacteria kind organism. Okay. And whole life was anaerobic oxygen. Life evolved, they're anaerobic life. Okay. You see that? So life came, so oxygen rose. So oxygenic photosynthesis starts around somewhere here, 2.5 billion of years ago. Okay. And you can see the oxygen concentration started rising, right? And you can see therefore CO2 consumption increased because of photosynthesis. So CO2 decreased on the earth, and methane concentration with anaerobic life flourished here. Methanogenetic bacteria, that is or archaea. Above archaea are methanogenetic bacteria. Okay, they produce methane, right? Methane, so earth are full of methane, and when oxygen concentrations became so high, okay, what blew your oxygen, right? So it became high. You can see, how methane concentration decreased. Okay, because aerobic life prospered, anaerobic life perished, lot of them, the methane decreased. Okay, there are many other reasons why methane was high and methane rose also. So you can see CO2 concentration has constantly decreased. And today's CO2 concentration is very minimum. Okay, that we had in in our geological past. But when you say CO2 increasing, we are talking about this right here, from here to here. Okay, do you see that? This is the CO2 concentration increasing we are talking about at the end, tail point. Okay, all right. So compared in our past, we have much higher CO2 than what we have today. Okay, where the CO2 gone? Lot of them became fossils, coal, petroleum. Isn't that it? They absorb carbon dioxide, a lot of carbon absorbed. Okay, then become carbohydrates in plants or animals. Okay, after they fed on the plants, and a lot also huge extinction events took place. Those extinction events led to what? Led to all them going to the earth bottom and becoming fossils from petrol or coal or petroleum. Okay, that's what we're getting today, digging from earth now. And now, therefore, we are burning them. So now what we're doing, digging those fossil fuels from earth, those carbon dioxide fixed billions of years ago. Okay, went underground and not available to us. Okay, now we're digging them out from the earth and burning them. So CO2 concentration is slowly increasing. Okay, and also cut down all the forests. 70% of forests are gone. Only 30% of forest is surviving. Okay, so natural no carbon fixation, so CO2 is increasing again. Not only that, temperature also increasing. Okay, because you cut down the trees. Okay, not only that alone, there are also other events take place. What I am saying, okay, so you look at rise of oxygen that took place around 2.5 billion of years ago. You can see origin of we are being origin of Earth 4.65 billion years ago. Life came around 3.5 billion of years ago. Around 2.52 after 2.5 billion oxygen was there, but it is 1%. Okay, there is 1% oxygen. And 2 billion of years ago came oxygen. Okay. Then of course one oxygen came, oxygen photosynthesis started. The ozone layer. Ozone layer. One have oxygen. So there are a lot of UV light penetrating Earth's dosage. So 3 molecular oxygen in help of UV light formed ozone. Right? And ozone went to the stratosphere. Okay. Then this prevented the penetration of UV light. Okay, UV light, and therefore what happened? It protected our DNA, generating material from damage. Okay, that hastened the process of evolution after formation of ozone layer. Okay. Right? All those prokaryotic life those days were haploid DNA. Okay, were getting damaged by what? UV light. Of course, they evolved a set of genes, including to repair DNA. Okay, DNA photolysis. Okay, but that repair could not compete with the destruction. Okay, so a lot of mutation took place. But that hastened so plants could not perish. They survived. Okay, and therefore evolution became faster. Okay, because they could survive. Okay, they are not damaged by the lethal damage by the UV light. UV light was there still. You look at some UV light. Okay, well, minimum UV light. And then you can say eukaryotes developed about 1.5 billion of years ago, right? Eukaryotes. Then of course algae, then development alga, higher animals, higher plants, vertebrates, invertebrates, all took place in the ocean. We are all talking about life in ocean now. nothing on land. Okay. So much life evolved in ocean. Okay. That the bacteria, cyanobacteria or algae and all that, marine algae, they converted water to oxygen. Right. Whatever they evolved in water, our life evolved in water. Right. Our life is very full of water, 90% water. Right. Of a cell. Why? We evolved in water. Right. Evolution started in water. So our life is full of water. Okay. So so lots of life evolved, and then eukaryotic sexual reproduction starts, animal develop skeleton, okay, and part came, then all came in life till only 400 millions of years ago, okay, came the land plants, okay. So till that, all evolution took place in the sea, ocean, okay. Now this came to life migrated to land. Came from bryophytes, then pteridophytes, then came gymnosperms. But when pteridophytes are growing, then came what? Land animals in form of amphibians. Okay? And then came amphibians, reptiles, and all that. So then came, long after came land animals, then of course reptiles, mammals came here, dinosaurs come in between, right, and reptiles, and they died. Of course, they become extinct. They become extinct because of some geological events. Okay, some collision of asteroids with Earth. Okay, so they become extinct. Okay, they're huge animals. Many species become extinct today. Huge mammals become extinct. Ramapithecus become extinct today. Okay, all right. Huge dinosaurs become extinct. But you can see original dinosaurs. Why I'm telling all this? Is to drive the point of important photosynthesis in our evolution process. Okay. When dinosaurs evolved, when oxygen concentration was 80%. Today, let's if assume today's oxygen 100%. Dinosaurs evolved when they are 80% oxygen. Okay. So dinosaurs are huge animals, not it? They have huge lungs capacity. They need a lot of oxygen for the survival. So their evolution had to wait till oxygen concentration in the atmosphere became 80% of today. Then only dinosaurs came. They became extinct because of extraterrestrial events. Not that oxygen concentration dropped or anything to that. Okay. All right. All right. So that I'm saying, how the oxygen derived from photosynthesis by photolysis of water in ocean to oxygen has driven the evolutionary process. Okay, and how then comes mammals, reptiles, then came birds and mammals, right? Birds came with the flying lizards, right? Birds are flying lizards, right? Then came mammals, right? Right. Then we came only 1 million years ago. So our origin is very recent compared to what has gone through. If you think our where we are today. Okay. All right. Okay. So we therefore we owe our evolution to those cyanobacteria who grew here. Okay. Acquiring photosynthetic apparatus. Acquiring chlorophyll A, bacterial chlorophyll A converted into chlorophyll A, bacterial chlorophyll, then of course the 3 oxygen evolving complex, 33, 23, and 18 kD proteins, manganese at the center, the oxygen evolving complex that adapted in process of evolution become oxygenic photosynthetic apparatus instead of anoxygenic photosynthetic apparatus. Okay, if this oxygen, let's look at temperature. We always link our evolution to temperature, but look at this. The Silurian age, 443 million of years ago. Silurian, you know, early pteridophytes started. Rhynia and all started in the Silurian age. Then Devonian, then Carboniferous era here. So you can see, you see a dotted line here? Dotted line? Anything to left of dotted line, left to dotted line is the cooling phase. Anything right to dotted line is the warming phase. So what you can see, Earth is warm all through, except during Carboniferous period, okay, and all through and right now, today. So today we are in ice house. Temperature of Earth is really low compared to what we had in geological past. Look at it, except Carboniferous. We had two more, ice house before this. That was not, but that is beyond evolutionary process, land plants, land animals. Okay, so I'm showing those. So these are two ice houses, means cooling phases. One was the Carboniferous phase, one is right now. And our evolution took place in this present cooling phase. Okay, in the last 1 billion years, Homo sapiens. Okay, so now it is a cooling phase of Earth. Okay. Now the temperature increase we are talking about or decrease talking about, you see here. You see, here is cooling, again warm. Yeah. Always cooling and warming going on. You see that? It's cooling, then warming. Cooling, warming. Cooling, warming. Cooling, and so on and so forth. So cooling phase we call glacial phase. Okay. Between two cooling phase we call warming phase we call interglacial phase. Okay, interglacial phase. Glacial comes from glaciers. Okay. Okay. Cooling phase is called glacial phase, and warming phase between two cooling phase now is called interglacial phase. Okay. So now we are in interglacial phase, warming phase, still a warming phase. But maybe last 20,000 years, okay, you see, all human beings came. When we made a society after knowing agriculture, right? We are all tribals, right? Moving in the forest, right? We learned agriculture. Only after we learned agriculture, around 10 to 12,000 years ago, okay, maximum 50,000 years ago, okay, then we made a society. We stepped together, one place. We did not move for sorts of food, right, from one place to another, right? So we settled down in one place. Okay, we lost nomadic life. Okay, and there we build our civilization. Okay, so that is the phase. This is the phase. Okay, so in this phase, last 10 to 50,000 years, our development is so high. We have destroyed all the forest 10 to 15,000 years ago. Okay, within the last 15,000. This is for human interaction nature. Before also we had human interaction nature. You see, one is geological event, cooling. So why warming and cooling? They all determine geological event on which you don't have any control. Okay, earth cools or warms automatically. That may be because of sun, how could do sun's power, and many internal things, cooling phase and warming phase that goes on all the time in Earth because of rotation of Earth around its own axis. Okay. Internal cooling is always there, internal cooling, okay, and then comes on internal warming. So the geological event that goes on and on and on all the time. Okay. So we might have destroyed so many plants. Our interaction nature is so devastating that we might have delayed the cooling phase so much. The cooling phase is not come yet. It has come by now. Okay, it's every 15,000 years, 10 to 15 years cooling phase, warming phase, cooling phase, then warming. You can see there. This difference are between these is around 10 to 15,000 years. Okay, we are already past. It is all still going on, warming and warming and warming. But you see going down the warming, but it's not going to cooling him now. So I don't know whether cooling will come or not. Okay, that's due to human interaction nature. Okay, it should come. It has started by now already, but delayed the cooling phase. Okay, the glacial phase is not coming. Okay, it should have come back. Okay. Of course, CO2 concentration increasing, temperature going to increase. Okay. Ice is melting, icebergs are splitting, glaciers are melting. What is the difference iceberg and glacier? Huh? That is the asking what is the difference of glacier and iceberg. Have you seen the movie Titanic? कोई देखे हैं आप लोग? हाँ? Titanic hit an iceberg. Where? In ocean. Right? So iceberg is in ocean. Okay. Glaciers are land mass. Glaciers on the land. Okay. So land mass, ice deposits are called glaciers, and ice deposit in the ocean called iceberg. Okay. Therefore, saying icebergs are splitting, see? And Titanic, and these glaciers are melting, polar bear is out. Yeah. So this is the university of life. Okay. Bacteria, then eukaryotes. Okay. Then the green gliding bacteria, green sulfur bacteria, purple bacteria, heliobacteria. Then came here cyanobacteria. Okay. They're all prokaryotic life. Okay. And this side eukaryotic life. And again plant. Okay. And of course, cyanobacteria became an endosymbiont, okay, and became chloroplast. Okay. This person who discovered archaea. Okay. So previous anoxygenic photosynthesis. What is anoxygenic photosynthesis? When it does not evolve any oxygen. What is the electron donor to reduce carbon dioxide carbohydrate? Hydrogen sulfide, let's say. It's hydrogen sulfide acetate, right? They are anaerobic photosynthetic bacteria. What do? They H2S plus CO2. CO2 become carbohydrate and sulfur. Right? Aerobic bacteria, cyanobacteria, you have water plus carbon dioxide and carbohydrate and oxygen. Right? So oxygen is getting evolved. Okay. Anaerobic bacteria, could acetate, could anything else, could hydrogen sulfide. Okay. They are okay, and they have one photosystem. Which one? Anaerobic bacteria. Okay. Whereas the aerobic bacteria, including cyanobacteria, have two photosystems. Photosystem 1, photosystem 2. Okay? And there are also two, I will not go, there are also two types of reaction centers in anaerobic bacteria. I'm not going to discuss about that. Okay. So, this is anoxygenic photosynthesis. right? That took place billions of years ago, around 2.5 billions of years ago. Okay. There came oxygenic photosynthesis. Okay. Billions of years passed through with anaerobic photosynthesis and aerobic. Between them came the intermediate transition form. It's called Chlorocoris marina and Geobacter. Example. They are intermediate type between anoxygenic phototrophs and anoxygenic phototrophs. What does the oxygenic photocyanobacteria did, and our modern day algae chloroplasts are doing? You do oxygenating water to oxygen and forming carbohydrates. Okay. Hello. Photosynthesis. Right. What makes a photosynthesis? Let's say, give me an example. Sulfur bacteria, purple bacteria, you know it. Many kinds of bacteria. Red bacteria, rhodobacteria. Right? You know, there is a which bacteria we had a first crystallized for photosystem crystallized. Rhodobacter capsulatus. Where photosystem were crystallized. We know the modern structure of chloroplast photosystem, bacterial photosystem actually in those days. Okay. All right. So, what is the Geobacter marina? Marina. Let's talk about that. This is marina. It doesn't have it has a chlorophyll D. Chlorophyll D containing organism. Okay. Bacteria chlorophyll A transformed to chlorophyll D. Okay, not chlorophyll A. Okay. What is this? Discovered in 1996. Okay. Now you also chlorophyll F containing organism. I'm not going to show you today. You have to see these are many forms of bacteria chlorophyll, right? Bacteria chlorophyll in red. You see that? You are observing around 780 nanometers. Chlorophyll D observing around 700 nanometers. Then comes chlorophyll A observing 653 nanometer. Then came chlorophyll B in observing 645 nanometer. In between divinyl chlorophyll A observing at 655 nanometer. Okay. Okay. If you see in the process of evolution from bacterial chlorophyll, anaerobic life, bacterial chlorophyll A, okay, we are observing 780 nanometers to chlorophyll D, which have observing 700 nanometers. Then came evolution of chlorophyll A, who observing 660 nanometers, and then came chlorophyll B in process of evolution, 45 nanometers. The wavelengths are shifted from red to blue. Wavelengths are shifted, right? From longer wavelengths, starting 780 to 645 nanometer in chlorophyll B. Okay, that's the process of evolution. Okay, and this transition from bacteria chlorophyll to chlorophyll via chlorophyll D, and of course we have the chlorophyll F. Okay, all right. This has promoted oxygenic photosynthesis. Okay, process of evolution. Okay, the transition. Okay, and what has happened? This anoxygenic photosynthesis bacteria. Okay, you can see here they have L and M subunits. Okay, we call it today. Today you have D1, D2 proteins. Same thing. Okay, in the process of evolution. You see that? And in between is the primitive reaction complex. In like a marina, we have chlorophyll D. Okay, same structure, twisting structure. No change in the billions of years evolution. No change in photosynthetic apparatus. Male photosynthetic apparatus we evolved in what? Long ago. Okay, I remain unchanged. Starting from anaerobic photosynthetic bacteria to aerobic photosynthetic bacteria. Okay, billions of years. Okay. In between, what happened? Recruitment of 3 proteins, 33, 23, and 18 Kd proteins in aluminum thylakoid with manganese center, having a manganese calcium oxygen complex. Okay, with 4 manganese there, which evolved oxygen, could able to oxidize water. Not only that, is supported by chlorophyll D and chlorophyll in process evolution, chlorophyll D or chlorophyll A. Okay, because chlorophyll A can absorb at what? Shorter wavelength, 663. Longer the wavelength, less is the energy. Shorter the wavelength, more is the energy. Energy of photon equals CH by lambda, wavelength photon, inverse proportional. Right? So what happens? So they got more and more energy to break water to oxygen. Okay. There are also other things involved. I'm not going to details. So they started evolving oxygen. Okay. And then evolution, what has happened only? The complexity process has increased. Same fundamental process remains same. Okay. Complexity increase. Same thing is Rubisco. Rubisco evolved in anaerobic world. Right? Right? Rubisco evolved in anaerobic world. Where is Rubisco? Photosynthetic bacteria, anaerobic photosynthetic bacteria. But we are not calling it Rubisco when we have carboxylase. Why? Because there is no oxygen there at that time. Right? So it did not have an oxygenous function. Anaerobic world. Okay. But as and when these 3 guys evolved, which one? These 3 red guys, 33, 23, 18 Kd protein evolved, okay, recruited, okay, and they oxidize water to oxygen, okay. When this happened, then there is change in the system. Okay. Then oxygen concentration went up. When oxygen concentration went up, the Rubisco which evolved anaerobic world. Do you understand that? Rubisco evolved anaerobic world. So it was not, so it did not see why it should protect against oxygen. Right? Did not evolve. There is no need. Why it should protect against oxygen. Right? So then it started compete. Oxygen started competing with Rubisco. When oxygen went up, then came photorespiration. Photorespiration came much later because of evolution. Became when you see when photorespiration came only here. Photorespiration here. You see, your oxygen is high, CO2 is reducing. Only when it reached somewhere here, photorespiration started. Okay. Also, the high CO2. So CO2 is outcompeting oxygen. Okay. When photorespiration became, oxygen concentration CO2 became very low. Okay. One of the reasons became flourishing of plants, of forest, lots of forest cover. Okay. Right. Oh, and lot of life in ocean. Okay. So around only 40 millions of years ago, Right? They're photorespiration before, but only 40 million years ago we got C4 plant. Because to counteract photorespiration, only 40 million years ago. Okay, why? Because plants could not or didn't change rubisco through the evolution of 40 million years ago. Right? Could not change the rubisco in favor of carbon dioxide against oxygen. Right? For millions of years. Therefore, the evolved came out with some other plants, other mechanism to concentrate CO2 in the vicinity of rubisco so that CO2 outcompetes oxygen. Right? The CO2 concentration increases, oxygen lowers, then CO2 outcomes oxygen. Right? So C4 plants, you know C4 plants? Huh? What have C4 plants look? C4 plants evolved in the to increase the CO2 concentration the vicinity of rubisco in the bundle cell cells. Okay. Mesophyll cells don't have rubisco. Okay. So therefore, the CO2 concentration increased, photosynthesis rate increased. What do you say?
Yeah, now we can.
Stop, you mean?
Your wish.
Okay, what's the time now?
1:45. Can have discussion.
Okay, all right. So we can have some discussion now.
I can sit.
Yeah.
Have a seat and.
We can continue tomorrow, maybe.
Yes.
Yeah. Yes. So let you complete 1 or 2 slides, maybe. So I'm not talking about that. Here you can see bacterial structures here. Why I'm saying this? The structures of bacterial chlorophyll A, chlorophyll A, chlorophyll D. Okay. So bacterial chlorophyll A structure, chlorophyll A structure here, CH2 CH3 group here. Chlorophyll B, you know, CH3 becomes CHO here. All of you know that. CH3 becomes CHO. But here chlorophyll D, instead of this CH3, this vinyl group becomes CHO. This vinyl group becomes CHO. Okay, CHO. So not CH3 becomes CHO, but vinyl group, that rate, vinyl group becoming CHO. That is chlorophyll D. So these changes have taken place. Wavelength changes. 752, 600, 700, then 663, and then 645 because of methyl CH3 becomes CHO. Right? That has taken place. What has taken only in the side chain ring, outside periphery of the macrocycle. Okay, basic macrocycle. Okay, having protopophyl ring with magnesium at the center. Okay, remained same throughout the process of evolution. Okay, only slight changes have taken place in the periphery ring of the ring, side groups, to change the absorption properties of the pigment. Okay, from 780 or 760, whatever, to 645 today. Okay, so this has been account. So therefore, I'm telling you, evolution is so conservative. You saw European not changing much. You saw rubisco is not changing much, except that some changes ratio of 8 large, 8 small, 2s, 2 large, 2 small. The many parameters of combination have gone on to the process of anaerobic to aerobic photosynthesis. Okay, but remains same. When the essential structure of photosynthesis apparatus I showed you, D1, D2 proteins twisted structure, the core, right, remains same. All right. Give me, okay, now discussion. Tell me, you see, you say chlorophyll have many kinds of chlorophyll 663, chlorophyll 680, chlorophyll 690, many forms of chlorophyll, right? Or not? Have you heard of that? The main forms chlorophyll in plants or not? Chlorophyll A is there. Let's see there. If chlorophyll A in 80% acetone observed at 663 nanometer, right? Or not? So same chlorophyll A, but when they are present in plants, are different forms of chlorophyll. Okay. Although while they are in such a broad wavelength, take algae, it does observe alga observe 663. Observe at maximum 678 maybe, or 680 maybe, a broad peak. Why is it due so? There are different forms of chlorophyll. Certain chlorophylls are bound to pigment protein complexes, thallic proteins, pigment protein complexes. Okay. Whether the head group is close to aspartic acid residue in the light hydrogen complex or a reaction center, near reaction center, or to a arginine residue. Arginine residue have what? Extra amino acid, an amino group, right? Aspartic acid extra carboxyl group. You know it. So they will have influence, negative or positive influence on the electronic cloud of the tetrapyral ring, tetrapyral ring. Okay. Their property changes depending upon when they are localized. Have you seen lobsters? देखो very give a very example. Lobster देखो blue lobster देखो. You, when you catch in sea, catch from sea, lobsters are blue. You steam it for a few minutes in the steamer, they go red. The chain of restaurants are red lobster. Actually, they are blue lobster. Once steamed, they become red lobster. Right? Why this happens? When steamed, they go red lobster. Because the pigments, okay, is mostly carotenoid type pigment, which bound to protein is blue in color. But when it is released from the protein, it is red in color after steaming. You see the absorption property changed after steamed lobster. Okay, so the pigment is released, and therefore it becomes red. So you can see how much absorption property changed from blue to red. Similarly, the chlorophyll molecules, where they are observed, where they are bound to the protein molecules, which environment they are, whether they are in hydrophobic environment, complete hydrophobic environment, or they are in close to hydrophilic environment. Even in hydrophilic environment, the difference of how much, aspartic acid they have nearby, how many arginine lysine they have nearby, that influences the electronic structure of the head group. Okay, and that in turn influences the absorption spectrum of the molecule. And the absorption spectrum molecule is signature of the electronic structure of molecule. Electronic structure of the molecule determines the absorption spectrum of molecule. Similarly, electronic structure of molecule determines the fluorescence structure of the molecule. If you are good in quantum mechanics, if you know the structure, you can derive the absorption spectrum of the molecule. Similarly, if you know the electronic structure, you're good in quantum mechanics, you can derive the fluorescence spectrum of the molecule. What you do, you measure it in spectrophotometer or spectrophotometer. Okay, that is measured. But that may not be actual spectrum. Okay, go ahead.
Sir, from Utkal.
You have Utkal married? Wow. So, on one line. Wow, good. How many of you are there? 4. That's good. What is your name? From Utkal. Wow. And you? Rao. That. Utkal. So you have 6 of you. Okay, good, good. And some are from Hyderabad and some are from anybody.
Hyderabad, no, but his both are from Goa.
Uh-huh.
Mishra sir student.
Okay. Mr. Sir?
No,
Sir?
no, Prabhat
Prabhat.
from Vizag, Tamil Nadu. From Hyderabad.
You are from Hyderabad. Okay. Anything you want to say or ask? Free for all discussion. Not necessarily here. Go ahead. Based on the architecture. Oh, okay, yeah. Why some parts there is granular stacking, some parts there are no granular stacking. Why PS1 is present in the periphery and PS2 in the granular region? I have a reason for that. There is a, okay, I need a blackboard for that. Uh, not here. Okay, but I can, but that is, there you can write also. But
You can write here.
can you do that?
So when pen, there only.
You can write there. Huh? You just turn up that. You can go to the PPT mode, PPT turn up. Then you go to, do you have blackboard by? Yeah. That's what many people don't use it nowadays. Now we forget also all the time how to use it. You know, if I can, yeah, you sister,
Come here.
give me a copy. Get a board. The whole touch. Note. No, they have write a, okay. Good. Oh, we will. Okay. Good. You see, this is thalacid membrane, right? Right. The another thalacid membrane. Okay. Thalacid membrane has a bilayer lipid membrane, right? Is the lumen of the thalacid. The lumen of the thalacid, right? The two thalacid membranes, they are separate from each other, right? Now, here you have what I told you here, PS1 here, and or PS2 is also here. PS1 is also here. PS2 is also here. Here also PS1, PS2, PS1, PS2, PS1, PS2, PS1, PS2. The two individual thalacid membranes. So you can see PS1 and PS2 are uniformly distributed throughout the thalacid membrane. Okay. But when you stack together, the structure changes. How? Let's What happens first will tell you. Can get another thalaccal membrane. Thalaccal membrane. So the thalaccal membrane 1, membrane 2, membrane 3, right? There are 3 membranes, right? Membranes. Now they are stuck together. Okay, not independent anymore, but stuck together. But here you will see PS1 will be here, PS1, 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 and PS1. So PS1, PS1. Let's say I'm writing 1 only now. PS1. Similarly, here also PS1, PS1, but here will be PS2. Right? PS2, PS, PS2, they're all 2 now. Right? So in stacked regions, we have only PS2. Okay, when they touch each other, they're touching each other. They are PS2, and when not touching each other, they're called PS1. Why is it so? When they're independent, PS and PS2 are uniformly distributed throughout. Right? You saw that. But when they're stuck together, PS2 is in the stacked region, and PS1 in the unstacked region, outside region. Okay, unstacked region, right? Okay, there is concept. Okay, I will not go details of the called Gui-Chapman theory. What is Gui-Chapman states you that if you take a beaker, right, and let's say a very let's sodium chloride solution here, Na, here. So what do you do? Sodium ion and chloride dissociate from each other. There is sodium ion. So plus and minus. Plus will be minus. Plus will be sodium. Minus will chloride. Right? They will distribute throughout. Right? Sodium chloride distribute form, and there will be distribute Na plus and Cl minus. Right? Ions are listed throughout the beaker. Okay. Now let's say you take glass rod coated with polylysine. What is polylysine? A polycation, right? Lysine positively charged. Okay. We coat with polylysine. Okay. So now you have a charged surface, right? Polylysine positively charged. Right? So glass rod is now polylysine coated, positively charged. At this point, what you will see, so this is a charged surface, right? Say distance. This is your difference. You will see all the chloride ions will be nearby. All the sodium ions will go away from there. Hey, all the chloride ions negatively charged will be attracted toward the where? Toward a positive charge. Polylysine coated chloride and sodium ions will go away from it. So distribution of positive charge around near the glass rod will be like this. Distribution of positive charge will be like this. Hey, that is chloride ion. This is sodium ion distribution. Right? Less and less sodium ion near the charged surface, and more chloride ion near the charged surface. Is okay? All right. Now, if you talk about PS1, PS2, they are not. Chloroplast is in a stroma. Stroma has what? Sodium chloride, magnesium chloride, all different salts are there. Not water, right? These are solutes are there, na? Charged solutes. Now, or as I told you, pH1, pH2 are negatively charged in as a whole. Okay, the net negative charge. Although they are because they have, although they are mostly hydrophobic core, okay, but they have also outside extension beyond the membrane. Okay, doesn't matter. So they are negatively net negative charge. But negative charge of pH1 is more than that of pH2. We depend upon whether you have more aspartic acid, glutamic acid, or more lysine and arginine. Okay, they are balanced. Right? Let's say pH2 has 2 lysine and 6 glutamic acid. Net charge how much? 2 6 minus 4. Isn't it? So let's say pH2 has net charge 2 glycines, 2 glutamic acid, and 4 lysines. So net charge is minus 4. Okay. Let's say pH1 has 2 glutamic acid, same 2 lysines, 2 lysines, and let's say 8 glutamic acid. So what will happen? Minus 6. Okay. So therefore, pH2 has what? Less negative charge. pH1 has more negative charge. So what will happen? You have sodium ion and chloride ion, right? To negative charge, what is the sodium ion? Let's say sodium ion, okay, in your cell. Sodium and magnesium, they'll go and bind to the negative charges at charge surface, right? They bind to it, right? They're close by. It's called charge shielding. They see and seal the charge. They are no more negative. If negative, let's negative charge, sodium ion go and binds, this surface become what? Neutral. Okay. So this is neutral surface now. Which one? When I add sodium ion, so let's say this is now sodium ion added here, sodium ion added here. So this become a neutral surface now. Okay. Chloride ion sequester themselves away from it. Okay. They are not coming out from there. Negative charge surface. Okay. All right. Now this one. Then there is another neutral surface. So this is another, let's say, this is another thylakoid membrane. That's also got neutralized by sodium. So this is another neutral. I don't know how to erase it, but that's okay.
So back.
So this is another one.
Very right. Very right. Yes.
Eraser is there. Eraser. Guns. Oh, good. They are more expert than us. Okay. नहीं पे कर लेते ठीक है फिर आप do again ना then you have to pencil okay 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 now you got PS to PS and everywhere, right? Now what happens? This is negative charge, this is negative, this is negative, this is, then you have plus, 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 this is negative, 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 then plus, plus, plus. So these two are what? Neutral. Okay. There is a theory called van der Waals interaction and Coulombic repulsion. Charges will repel each other, right? Like charges repel each other. Okay. There is universal van der Waals interaction between two surfaces, whatever you see, are solid, liquid, and gas, whatever it is, because van der Waals interaction mostly. Of course, they have a hydrogen bonds, but they have van der Waals interaction in all nature. Okay, have two attraction. We'll not talk about the theory of interaction right now. Why is it there? Okay, you can. I'll talk. It doesn't take much time. Anybody don't have time to talk about that. So you have Van interaction. So these two surfaces, which are now uncharged, they get charge shielded, no charges, right? Neutral charges. Now they come together by Van interaction. Okay, so there is a grana stacking. Okay, stacked to each other. Okay, so all these neutral surface now stacked from the grana. Then why PS1 and PS2 are different? Because now PS1 I told you more negative charge, PS2 less negative charge. So PS2 will be preferential shielded. Who will shielded first? PS2 because less negative charge. Who will shielded much later? PS1. Okay. So now PS2 also negative charged, was negative charged. Now they are shielded from by sodium ion. So let's say you have 10 PS2 in a membrane. It's a membrane, right? Right? There are many PS2 here. Okay. Now PS2 are mutually repelling each other. They're all negative charged. Now they all become neutral surfaces. Now they'll come closer, aggregate. So they'll aggregate to one place. Okay. So this is a neutral surface. This is also a neutral surface. All the PS2 therefore will be together. Okay, stacked together. Okay. PS2 coming to the neutral surface. Because these PS2 were repelling each other. Now because sodium charge shielding and we have Van der Waals interaction between PS2 will come closer. Okay. And then there will be PS2 in one place here, one membrane. Another place, there will be another PS2 in another membrane. Okay. So you give me another one book. Yeah. The other membrane. So this is neutral now, PS2 zone neutral. It's PS2 zone and neutral. PS1 is not come together yet. Only PS2 has come together. So PS2 and this is PS2 here. So they will now charge their Van der Waals interaction. So these two membranes are together. All PS2 rays will be in the tagged region. Huh? PS1 will not come together because they have enough, so much high negative charge that the sodium chloride cannot shield them. Okay? Or magnesium cannot shield them. So they will remain in the peripheral zone. In stacking zone, they will be PS2. Okay. Thank you.
So we cannot hardly be here. Oh, very good, very good discussion.
All right?
Is it yours?
Yeah, you mind.
PMSF. 2 only. This is 2, right? One more is there. No? PMSF.
In that status.
Okay. Yes. Okay, let's start thylakoid isolation. You people must be tired of learning so many things, right? Okay, so we'll do thylakoid isolation. So we started from basic experiments with cells. Now we will go a little bit more deeper to study thylakoid membrane complexes. We will do tomorrow native page to see PS1, PS2 complexes separated and all. So if you want to separate, if you directly load the cells or directly you can't get the protein complexes, you need to first isolate thylakoid membrane. Then only you can proceed further experiments if you want to go deeper about thylakoid membrane proteins. Especially we will be focusing on photosystem proteins. Then for different organisms we have many different kinds of thylakoid isolation methods. We are here using a normal method using a tabletop centrifuge and sonication as the cell disruption method. There are many other ways to do. If you are working with plants, you can do normal grinding. When you did RNA isolation and all, you might have done the mortar and pestle. This you can do. You can use a grinder to extract the first crude extract and all. Then another way is using for algae, you can use this French press machines are there which works on like liquid nitrogen. It will grind with beads and all. Mechanical disruption you can do. Then another way is nebulization, which is like in high pressure you can pass the algal cultures so it gets disrupted. So we will use a common method which is sonicator to disrupt the cells and all the membranes will be out like in the solution whatever the buffer you are using. Then you use a normal centrifuge method to get the thylakoid. So in this method when you use a tabletop you are getting majorly broken chloroplast rather than a pure thylakoid. If you need a pure thylakoid method, you have to go for ultracentrifugation, like very high speed centrifugation method. That also you can keep gradients of sucrose so that the membrane will come in between the gradients. So those will be like the purest form you can get. But for our experiments, this is also fine if you do broken chloroplast and you can proceed. So majorly today we will do algae. Plant leaf, no, today we will be doing it the algae. But basically if you have a protocol for thylakoid isolation plants or whatever, you can follow it. It's not much difference will be there. Basically this all will have a buffer system to maintain. pH 7 should be maintained entire. And the other thing will be everything needs to be done in ice, or the best method is using a cold room. If it's a dark cold room, that is the best method you can do. You get the more stable and the membranes will be more stable rather than doing in a room temperature. And there are several protocols you get for thylakoid isolation depending on your organism, whatever it's a monocot, dicot, it's an algae or bacteria. These protocols will change little bit here and there, that's it. Basically you will maintain a buffer system, and also major thing is you will use calcium or magnesium components, MgCl2 or CaCl2. These both Mg and Ca both are in the system itself, photosystem itself, so they need to stabilize this. So we add magnesium and calcium, and there will be sucrose which is just to maintain the osmoticum. Once you are disrupting the cells, they are going to be in a like they are going to burst if you first wash itself. If you don't use a sucrose or just like an osmoticum balancing, you will use sucrose also. Then you can most of the protocols they use HEPAS as the buffer for maintaining the pH. pH 7 to 7.5 will be maintained in most of the protocol. These things are common. So if you want to
also you can put it in 2 to 8 degrees.
For initial pelleting, you don't have to keep it in 4 degree because they are already in a room temperature and we are not disrupting them. We are just pelleting them. So they are okay with 25 degree till. So it will be 25 degree we'll keep. Once we start disrupting their cell wall and everything, then everything should be in ice. Or you should take to cold room.
Okay.
Dark and cold is the basic,
Face problem, actually.
but we do like that only.
Protein purification.
Everything in cold room, we have to stay like 2 hours or 3 hours in cold room and we have to do that. That is the best way.
You are using octaprime or octaprime? Octaprime you are using, or directly can how you will to purify the protein?
No, we are not going to do that way. We are going to isolate the protein complexes, no?
Okay.
Using gel methods.
Okay.
Blue native page we are going to. You are talking about the column purification. We are not going to do that now. And that level experiment we don't do here. Like if you need pure PS1 and all, you can use this column purification and all. Yeah, you can use. But we are right now not doing that method.
But anyhow, you are non protein. That's why how to.
Yeah. We are not going to work with any unknown. We are going to work with known complexes. And
You'll be isolating thylakoid proteins. Or just
we will be thylakoid membrane.
proteins.
So obviously thylakoid membrane, the protein complexes will be embedded in the membrane itself. So basically we are isolating the thylakoid membrane proteins, not in dissociated form. As such complexes we are going to isolate.
Can I use the same for later my clotting experiment also?
You can use. And when you work with photosystem proteins, it's always better if you use thylakoid membrane proteins. You will get more, like you can do, instead of protein estimation.
Isolation protocol will remain same.
Yes, same. You can use this extract for several experiments, not only for.
The organelles.
That's what. If you want to get pure form, you have to do gradients of sucrose.
But anyhow, you are doing pure form. Actually.
This is actually broken chloroplast you will get now. Whatever the centrifuge we are, the 10,000 we are keeping, the broken chloroplast will be pelleted. But in that also there might be contamination of other membranes and all. If you go for an ultra centrifugation, you will get the topmost like pure form of thylakoid.
Able to extract the specific PS1, PS2
Yes.
by using of the thylakoid.
Yes, you can. You can do sucrose density gradient using this thylakoid membrane. After solubilizing the, like, it's a membrane, now proteins complexes PS1, PS2, cytochrome B6F, everything will be embedded. If you just solubilize this membrane, this complexes will come out. So, and if you solubilize this, basically called solubilization. We do denaturation for SDS page. So here we do solubilization method, so that the protein complexes will be intact. The proteins will not get denatured. They will be intact complex, but only it will cut the membrane. So membrane will be cut, PS1 will come out, PS2 will come out, and you use different detergents for different complexes. If you want to work on PS2, there will be specific mild detergents which will help. If you want to work with PS1 more, you can try other mild detergents, not SDS. SDS will denature everything. Every protein, everything will be denatured. Here you will use only the membrane specific, so that the complex will come out all with every subunits. It's a multi-complex. Now you load it into sucrose density gradients, and PS1, PS2, LHC2, everything will separate. You can take each fraction, you can do further purification steps like column filtration and all, and you can get pure PS1, PS2, everything. For degrading. Yes, there are several detergents. Yeah. Depends on, like, grana is more stacked. So some and you know the distribution of PS2 and PS1 will be different. So according to this, if you want to focus on stroma lamellae, you will have mild detergents for that, and for the stacks, which is like more stacked, you need a different micellar which band is what and all. So we know now already. That's it.
difference between blue native and SDS page? Because in SDS page, because in SDS page also we can get the protein.
Yeah. So the difference is basically here you are going to work with the protein complex. In an SDS page, you are going to work with the protein, individual proteins, and also individual proteins you will denature, heat it, and make the primary structure that is going to separate. Here you are going to work with the entire complex. So you should not cut the bonds, disulfide bond or anything. You are using basically SDS and all for cutting the disulfide bond, all this, and make it the linear structure, and that is the denatured protein. You don't want to work with a denatured protein here. You want to work with the functional. Want to work with a denatured protein here. You want to work with the functional complex. And when you isolate using SDG or whatever, they are functional complex. Even you can work with adding something. Their redox kinetics and all we studied. No, you can isolate this pure complex and study the redox kinetics is also. They are functional. So that is the difference why what is SDS and blue native.
In blue native we get the entire complex.
Yes.
In SDS we get individual proteins.
Yeah. Yes. There is no denaturation in blue native. Got? Can you bring something to discard this? Is that?
No, it is for medication.
Okay. Ah. First one we will be using CRB. So let it be nice. And what about light? We will do in dark or?
But you switch off it. We can say it.
No problem.
Till thylakoid, no light, not issue. Medication,
Maybe we won't expose, yeah. We have pipettes. Maybe 2 we can take. One more, then we'll resuspend it fast. So you resuspend directly in TRB1, right? No, CRB, right?
direct. Make it 20 or something. Find a
Can you take one more set so that we can do? So this is the first buffer, which is called CRB buffer. Basically contain the like sucrose, HEPS for maintaining the pH, and basically CaCl2 and MgCl2. That's it. You are just going to wash the cells. Any other particles or something is there, we will just wash, and the excess media will be there, whatever it grown. So we will remove, and the media will have another ion particles and everything. So we'll just have a washing step.
CRP buffer. Pavitra? CRP buffer.
CRB buffer. I think in protocol book we may,
shared already.
right? I don't know.
Which one is it? I didn't.
Yeah, even if you just type chlamydomonas thylakoid isolation, there are most of the papers will follow same. You will get, and you will have the.
Okay, I will do this side.
Yes. Maybe 2-3 are there.
If you are using a plant buffers, will be different.
Yeah, a little bit different it is.
Grinding.
Huh?
For plant grinding.
Plant grinding also you do, and buffers also little bit changes will be there. Because these are like single cell. You don't have much things to remove and all. But when you deal with a plant system, it will be more complicated, right? and you will have extra steps to avoid that crude thing and everything. Should I give someone to resuspend or I will? I'll give. So someone you like can do the resuspension.
You have to add 1 ml of CRP pepper and slowly make it to solubilize. There should not be any particles.
We take this. You can take both our CRB.
You do wear gloves.
Yeah. Any experiment you should wear gloves, but for now it's fine.
Sorry?
CRB, what it is?
Cell resuspension. Resuspension. So only 1 ml.
Only 1 ml and just resuspend it all. And for thylakoid isolation, whenever you work, you should work, I already mentioned, dark, cold and gentle you should be. And fast also. Because if you keep more time outside, you are going to destabilize the membrane. You are going to disrupt the proteins. So they should be intact, the protein. If you change something also, they will form some aggregates, they will denature, temperature, whatever. So you should be in dark, cold, very gentle, but very fast. If you have in institute like cold rooms, better to do there.
Sure. It's already shared. Shade. Look. It's in the good. Check it. GIAN.
When you do in cold room, I can't wait more than 10 minutes.
You should you can wear some jackets and you do like 3-4 hours continuously.
Yeah.
Whoever work with photosynthesis, thylakoid, they do this.
Protocol we shared in GIAN.
Yes, soft copy.
Yes, I remember I only shared GIAN.
So now we will pool and make it up to 50 ml.
30.
30. Okay. We need the tube. This tube only we will use, or we will use.
For what? Centrifugal or high speed?
Okay, now we will just wash with 5000 only. So we will take Falcon. Where is Ragin? Or a Falcon tube, 50 ml.
I think it's done.
Wait, I'll see. Yeah.
So everyone just check your group in GIAN WhatsApp group. The protocols are already distributed. So make sure that you follow that. Before coming for the next day experiment, you just read about it so that you will have some idea. Easy to follow what is meant.
And when you do like in a proper like experiment, you keep everything in ice before only. Maybe we will pour a little bit CRV. We have enough, right?
Yeah, 30 spots.
Mm.
Got 30.
No. It's still 20. Little bit
Otherwise,
more.
we'll make 60 divided into 2 tubes. So certification also is.
Yes.
Let's divide into 2. Let's dilute it.
I'll take half.
First make 60 so that concentration will go. Or
No, now it is
30 already.
No,
Okay,
no.
you made 50.
So depending on how much pellet you are working with, you can vary this buffer, how much you are adding. Right now I got like the pellet you saw. So we are just making it 2 tubes of 30 ml with CRB buffer.
This one 30. Okay. Yeah, exactly. The name is cell resuspension. Resuspended, so that the pH, everything, because immediately from the media you are transferring. So it's like giving, cells should not face any osmotic stress.
We'll keep that.
Yeah.
Normal. Yeah. Just no. This will be before sonication.
Now we need to sonicate.
No, washing step is not there.
One time we can, but you have CRB,
Let's check that, or else we can pull it into one.
Yeah.
So you skip this step.
We can skip. Yeah, finally also CRB.
Let's see. CRP, this much is there. This is there.
There means we can do it.
I'll do one washing.
Fine.
എന്താണ് ആര് നല്ല പിന്നെ
How many times you need it?
maybe 5,000.
How many times? Just once?
One more high you need. Right now I will not use, but we need to keep it for 4 degree. Maybe you can use in another lab for now. Don't keep like this, it will again. This is already pelleted.
Yes.
Then don't keep like that, no?
This one I'll do again. I do have some issues there with this.
Okay.
2 times. 5,000 only?
5,000.
What?
5,000 for maybe 8 minutes. ഇത് അപ്പോൾ മേ ബി ചെറിയ ട്യൂബിൽ ആക്കാൻ പറ്റുമായിരിക്കും. സോണിക്കേറ്റ് ചെയ്യാൻ എൻ്റെ കയ്യിൽ വേറെ ഉണ്ട്, ഇത് വേണ്ടി വരില്ല എന്ന് തോന്നുന്നു. അവിടെ വെച്ചോ എന്താ മോനെ? നിങ്ങൾ മറ്റേ സസ്പെൻഷൻ ബഫർ യു വിൽ കീപ് മൈക്ക് വെച്ചിട്ടാ.
per gram of
Decide product.
product is what you are looking
No, I don't know. That way I don't know how to tell it.
How do we know that it's well suspended?
Just if its clumps are not there when you do, it's okay. Just take and keep like this. If no clumps are coming, then it is fine. Normally you should do all the steps and all fast. We'll make like 25. Okay. You do the sonication. No, I'll add the inhibitors also. Again do 200 microliter.
Major thing is cell should not be clogged, otherwise your disruption process will not be effective. It's at least 60 to 70% cell should break open. If they are clumps, then the efficiency of the machine will not be that much. So try to resuspend properly. Be faster as well as be more efficient.
The tips. No. This is. I think 120. So you add all the inhibitors like 10 millimolar maybe.
Yeah, because cell open then as you are stating now. Once inside the cell, everything will be in the control. Once they come outside, they feel that they are in stress, they start degrading. Even RNAs will do the same process. That's what why RNA isolation, we should be very careful. Vanadium we add for RNA isolation, that it will be RNase. So similarly here we are using protease inhibitor.
So, because or we can use antibiotics?
We can use this. I use this only. No, I break it because if it is too long, the probe won't go till down, the bottom. So the cells in this area won't get the sonication power. So I just cut so that it will be probe will be complete.
10 millimolar. Three
Which one?
protease
Inhibitors.
inhibitors we are adding. One is benzamidine hydrochloride, and then aminocaproic acid, and third one is PMS. Benzamidine hydrochloride, BHCN short form. Benzamidine hydrochloride, aminocaproic acid, third one is PMS. Yeah, we can
No.
make
We can make cocktail, but right now I just added everything different.
What is PMC?
PMSF. Phenyl methyl sulfonate something is fluoride.
Yeah, you can. There is no particular protease inhibitors. It's up to you. We follow a particular paper where they use this and they found it's working better. So we started at
Everything 10 millimolar you add.
protease inhibitors.
How much they gave? That is the stock. I am telling the working 10 millimolar you add. This is I think for 1 molar. So from that stock you can add only 10 millimolar.
Protease. Protease is an enzyme that degrades protein.
10 millimolar means what? It's the concentration I am telling. So whatever volume you can add 10 millimolar according to the volume. This is too much. Put it in this.
It's like a rocket.
Use this
No,
one.
this
This or this one. But this is too
hard. Fine.
They filled it. Should work. It's still not straight, right?
$30 right?
Yeah, inhibitors added. Now you can directly keep the amplitude and all ready.
Yeah, makes sense.
So whenever you do, you check papers, some previous paper, what is the amplitude and pulses they used, and follow that.
So-
Now for clammy, 25% amplitude with a 15% of sonication on and 45 second 45 seconds on off cycle. If you give a 7 pulses like this, it will be properly done. This is like standardized. So depending on your organism, or if any papers which is related to your organism, you can follow that.
15-second on and 45 seconds.
45 second off. Off cycle you can reduce to 30 maybe.
If you want, I can write.
Yeah, reduce, no problem.
I thought something happened.
You just keep the probe inside,
So
come.
this is how we set the program.
This is the
This is a
sonicator
program
probe.
for one
It
minute.
go inside till the bottom.
45 seconds. We can
Just
reduce to the
sonication.
pulse to 30 seconds.
Because
It's on. Pulse will
everything
be
is
given
going to
only for
open
15
up.
seconds. If you continue
So-
to give, it will affect the protein.
no, ice temperature is best.
So 15 seconds we are giving the pulse and 30 seconds we are switching off the pulse. So the cycle will be repeating for 7 minutes. Until 7 minutes this cycle will start. So let's start. Can hear me?
Temperature also. What about the temperature also setting?
You set 4 degree and we are going to keep ice.
Not
So
means that is kept as a program in order to check whether the sample is getting heated up.
sample is getting heated up.
Fine. Until unless ice is there, no problem. And if you are going for a very long time of
Then the probe itself will be heat up. Then.
When starting, we have to do the dark condition.
Now from this top onwards, now the membranes are exposed. So if you expose it to light, they will absorb. The proteins, everything is now outside.
Yes.
So if you expose to light, they are going to change their assembly. Sometimes they will aggregate. They will do like if they get light, they get excited. So it won't be the natural form. So that's why you should do it in a very dark light, or you can use green light also, which they doesn't absorb.
Like dim.
Yes.
Seven pulse will give automatically.
No, probe normally don't get heat up. But if you keep a prolonged time for sonication, the probe may get heat up. So ice is for the cells are now going exposed. So the protein should not be in room temperature. Everything should be in ice for the stability. Otherwise they will degrade, no? So on, now they take an off cycle. Like this 7 times it will go. This cycle will be different, this amplitude will be different for different organisms.
Yeah, it's automatic. If you want more power, we generally don't go for.
For breaking the cells,
Yeah, we also
for breaking the cell sonication, then you can use nebulization method, which is like under high pressure.
Using of
Under high pressure,
okay.
there is separate instrument for that, nebulizer. Sonication, then French press. That is mechanical disruption.
This is like very convenient method. Just 5 minutes if you keep now. So people prefer to use this. But those things and all, I'm not sure how.
They are telling like sonication is more harsh. Nebulization is more mild method to deal with thylakoid. But it depends again. We always use this only.
Anyway, for plant people, they use grinding. So
it's again different.
So algae, can I lyophilize and then you get resting?
Lyophilization is not.
No, like some algae like no stock.
Can
I lyophilize the cells?
I don't think lyophilization will work. It extracts the water content and everything, right?
Maybe like RNA.
So it should not be.
Directly by liquid nitrogen.
But thylakoid,
I'm not sure,
no.
but we haven't done it.
Both of nostoc, many experiments first and lyophilize the cells and
Yeah.
then do extraction.
But thylakoid we don't do. Thylakoid we don't do. No. Mostly we do.
And these experiments will be done to compare how the photosystem complexes are with different samples. So generally, if you're going to lyophilize, as she was telling, the water content will go, and it will change the status itself, the native status.
You can't. Lyophilization you can't do for thylakoid membrane isolation. No.
Any other method to open the cell?
I told now 3, 4. How many more you need?
Any other like grinding point?
You can do grinding.
Grinding normal motor vessel.
For algal cells, no. No, it's more stress. Like you won't get it. It will destabilize everything. Homogenizer, what is that? Like
It's like a long rod.
that is just no. That won't help for disrupting the cell wall and all. I think no.
Can cells be disrupted using like high voltage and low voltage AC?
No, I don't have any idea with this voltage. No.
I think you buy. Just go to your map. The map was that very clear.
Mechanical will be too harsh for algal cells. It's not preferred. How many more?
2 more.
We can take out. Maybe at least this light we can switch off. Whichever it is, the switch.
You can switch off the lights.
At least one light we will switch off and keep it a bit darker.
Some samples they still outside.
Yes. What happened? Is it
No, I think it's.
leaking?
I'm not sure. Let's complete
Take.
for last one. Maybe while bubbling it would have come out.
But we have this. But still how? I kept 20 only, not 25 also.
That's it. Is it sample? Yeah.
The road was touching the bottom?
No.
It was bit up, right?
Yeah, bit up.
Then what?
Maybe while pulsing it would have.
But it should not. It just vibrate, it won't do.
Bubbling? No, bubbling out.
Okay, that's fine.
The sound is
We lost too much. Green ice you get. Let's see. Oh, I never used this one. It happened earlier?
No. Maybe
No,
for
it will be like this.
Might be you can reduce it.
Yeah, maybe.
Let's reduce it.
And now you will do the fast.
Yeah.
So I am keeping in this, whatever we got. That is fine. Maybe you can use.
I'll
Okay,
do start
wait.
But it will transfer.
Yeah. But very little we got because of the spilling. Should we use bigger one?
Bigger one.
Why it is happening? I don't know. It's not normal.
This is one of the error. What is this error called?
I reduced the amplitude. Let's
I
see
kept it 20 only actually, I think. 20 or 25. We'll keep 20, but
20.
still.
I kept 25.
Should we keep half?
whatever it, right?
Yeah.
But sonicated.
Yeah, it is sonicated, but
Spilling is
we will get very less thylakoid.
Okay, let's try half.
Half, yeah.
15.
Maybe. Let's see. It's the same fine.
Just hold this stop. Drop it.
Yeah, it's dropped. It's going. Keep the little space. Little bit, it should be
Yeah.
fine.
Check it once.
Okay, I will check. It's
Leave that.
stable now?
Yeah, leave that.
You do this.
Yeah. That's there. Enough
It's
space.
there?
Yeah.
Okay. Little bit.
Just now. See.
Okay. Yes, that is enough.
Because we are right, they don't see they have teaching and everything.
I reduced
If they are putting for research, then they keep
it's there. Giving more result.
Mm-hmm.
Now I think it will not change.
Mm-hmm.
your method.
We'll collect in one.
We'll make everything one.
Yes.
Okay.
Because it's less only now, no? See.
But it's better.
Okay.
Or we can half we'll make like this.
Depends. Let's see.
Yeah,
Also we need to do it.
Because anyway balancing you need. So let it be half. So cramming is very simple steps. Just sonication. And take the pellet.
Then we need to use the brush.
Yeah.
2 times we need to centrifuge. One with the hard.
Then
Yeah,
they
you
can
want-
maybe make ready the 77K thing.
Now?
What is that? Just
No, it will
I don't
take
know
some
how
sufficient
77K.
time. Now 7,000 is super.
Mm.
The resuspension takes time.
Ah.
There, at least
And this is 10 minutes or 20 minutes?
10 minutes, I think. Just check the photo. No, before. That page.
She knows. She already
Memorized?
memorized.
TRB one shit.
13,000, 10 minutes.
Sonicate, take supernatant, centrifuge. First low speed. Just to remove the debris.
Okay. Take supernatant and centrifuge it. Okay. Shift the pellet a small hard tube. Okay, there is another one more step.
Yeah. Initially, we need the sonicated cells will have debris. Debris need to remove. For that low centrifuge.
So we could have used this one, no?
Yeah, the same. We can use it.
We don't have to use this.
Yeah.
This we will use for the final step. So here we have a TRB1 washing.
Yeah.
But again, shift the pellet. Okay.
Means after the debris removal, we need to shift to the hard
To
tubes.
the, And there you use the TRB1.
Yeah, TRB1.
So that it will be pelleted.
What is the time?
Why? It's not going.
Bubbling something.
Bubbling is there, but the sound is not coming. Sonication sound.
Pulse off only now. Let's see next pulse when it starts.
Pulse off and it's vibrating. Why? We buy all the bad machines.
Any alternative is there if this doesn't
Again, come here.
This will come.
work? Alternative, this doesn't work.
Again, we need to do
It's
sonication.
positive.
We don't have. We do only sonication.
Some double.
Why this is sound is not coming?
Not coming. See?
The off stage mode.
Huh? No.
Last pulse also same thing
Yeah,
happened.
it was not giving a harsh pulse. Sound is not there. But only sound is not there, but bubbling is there.
Why?
And that is one.
We'll switch off.
Switch off.
Yeah. Stop. How many pulse it went properly?
2 pulse.
2 pulse only. At least 2 more pulse we need. Take out and see what is going on. Why it is not? See again.
This is like bubbling. What?
Too much.
Means not lost, I think. It's lost. Oh. Can I get a pipe?
Yes.
No, first time.
അതെന്താ മനസ്സിലാവുന്നില്ല. ബബിൾ ഇത് പുറത്തേക്ക് പോകുന്നു. വലുതാ എടുത്തത്. Okay.
So because of this,
ഇത് maybe we could have used a bigger one.
still there.
It is there,
Yeah. But it was bubbling. I think pressure is too much. The power is too much.
but maybe
You use the same probe but small probe. Same probe only.
20 amplitude. Amplitude എത്രയായിട്ട്? Amplitude 10 only they can. Maybe for this probe it is different, so we will stop this. We'll take this because in 2 pulse itself it must have done.
Maybe it's giving high voltage.
Yes. This is bigger
Yeah.
than normal.
Okay, then I will. One more time is there.
Yes. Keep amplitude 10 and see, maybe simply.
Okay. Maybe we suspect the power rate is high. That's why it's the whole probe is being. So I reduced the bone power. That's it this time. But anyway, the purpose is done. Only thing it's giving lot of foam.
When I do nebulization, no, it will be full of foam.
Foam. So you did it. Oh, you do nebulization.
Nebulization there. I showed no that day. Not here.
No, I don't think so.
I showed.
Ah, good.
I showed the machine.
Okay.
That is the one. I did in that only. I did sonication also. Keep normal temperature.
That give you shock to that particular sensors.
High pressure, you will pass something. What will happen?
Number 4. Bursting.
Bursting. That only.
It's similar to like how you put it in salt solution.
Now we are trying 10. Maybe we need to use that wider
Maybe.
This tube.
or wider beaker കൊണ്ടുവരുമോ? Wait, we'll transfer it to a wider so that the bubbling and outside it won't go. Let it be there. She will bring. No. Next step we will centrifuge it and you get like the supernatant, you will have the membranes.
Okay.
And down all cell debris will go.
Okay.
So this membrane you will take and you will add another buffer
Okay.
which will make them the membranes to pellet down next in a high centrifuge speed. 12-13,000 you can use.
Okay, now when we scan the
BN page we will do day after tomorrow.
Scan.
77 Kelvin.
Okay. We'll do right after this. Plant leaf, you use a normal grinder, mixer grinder.
nothing will happen. It won't damage.
No. It won't. No.
You open up the search. Now to remove the
Use this one. Jero,
Mixie also it can.
maybe we'll use this one. Let's see, at least for your future work it will help.
Mixie also you can use for that.
Ice with
Mixer, you can ask him. Like he use mixer grinder for plant. And they use, what is that cloth?
Muslin cloth.
Muslin cloth they will use, yeah.
Okay, muslin cloth.
Yeah. Filter and take the crude. Then you will start adding the buffers that we are adding now, no? Like that you will add and you will pellet them.
Procedure is
ഇത് ക്ലീൻ ആക്കോ ഒരു ഇതെടുത്തിട്ട് വാ ട്രേ.
even LH is also like the same way that the
Which one?
light harvesting complexes.
ആ
Procedure is same like this one. It is also pretty.
I didn't get the question.
LH light harvesting.
Light harvesting complex.
That
In
is
thylakoid membrane you are collecting everything, right? Thylakoid membrane, your LHC, everything will be there, right? Maybe this also, but I need this right back. What is the situation? Now it is, sound is also fine, right?
It is fine. Let's see second.
This is done one pulse already.
Yeah, one pulse now only. First pulse.
Oh, this is first. And 10%.
Yeah. 5% I reduced to 5.
Nothing will be there now.
No, I think this is powerful. This is very strong.
More power.
That's why foam is, this much foam is forming, which I think they are very harsh.
Very harsh, maybe. Fine.
Second one, some
Hmm?
it is off. Huh?
We are
Now it's
actually
off.
experimenting. Let's see. We are also like you. I think it's working now. Maybe because of the
For that probe,
wall is
28
very-
might be more. I thought you already used with this sonicator.
This sonicator is
Okay,
recently came.
recently.
Only Raghendra is using right now.
Uh-huh.
It's for demo purpose. We just start.
Mhm.
Know how to use that.
Mm-hmm. And we need to do the chlorophyll estimation also. Just-
Alvin, though, you can directly go.
No, anyway, I need for BN page, I need at least some idea, otherwise it is difficult. It's me. This is my first year book.
For 5 years, they give 5 books. You should complete all the 5 subjects.
No. How much you want to write, you will give. They will give record. How much work you do.
Every week correction is there.
This is your just record what you're doing and all.
Like a diary. Like
Laboratory diary.
but very formal.
Two is done.
Okay.
4 to 5 is enough. I think this is the best. This group. Raghendra also use this. She said. No?
She used 10% probe, like amplitude. For 1 hour they are keeping
But the same container.
same container.
Maybe hers is very hard, sir.
Yeah.
That's why. But that is nicely set. That is nicely sonicated.
Yes. Let's see.
Whatever the records are mentioned, they will present them, including the buffer composition and
Yeah, it should be.
and
Oh, okay.
all these experiments.
Yeah. This is the once you go back, you just try to do by yourself. Write it again properly.
And thinking whatever the sources you have and proper.
Yeah.
Yeah.
Sorry.
What is?
Here in this university,
What?
there is any portal for using common facilities for outside students.
Chemistry it is there. STEM is chemistry, I think. Department of.
STEM is like we know list of instruments where we can use.
Maybe I'm not sure. I never.
You want to use sonicator?
Don't get them right.
Here how it is. For chemistry department we can.
Here I think department.
Which one? See that?
in science
Previously, any outsiders have come and done
So
an
if you
experimental
go there,
work
they
here.
will show what are
Yes.
the facility which institutes are
One of my
providing.
objectives is entirely photos.
Mm.
So whatever experiment I have done,
Mm. Mm.
I want to do. So in case he doesn't proceed into my so I
You can talk to sir. He will help.
here we are institutions like there you get like exams or those things. Then they checks in and something like that if you want to do something you get an exam like that.
I think in university that common portal is not there.
They have system. It's like common. Wherever, whichever institute.
Where
it is.
university or there you
No, it's like common.
also you are grinding that green thing, right?
Always wipe the probe.
So
No,
same you will get smell.
nothing. It's the empty cloth, tissue paper, or with water. That's fine. No solvent should be used.
I need this cap.
There, it's not there.
No.
Okay, use it. The one unused. So I think work is done now. Can you
Yes. So let's go for.
send it?
Should I add more CRB?
How
It's
much
too
it is?
less.
Okay, add it. Okay. Make it 30 30.
Mm.
if I'm doing this right from starting in cold room,
Mm.
right from starting
It is the best way. You should not do like what we are doing now.
plant, then starting from grinding onwards.
Will be good. I always suggest everything to do in cold room. That give you the best result.
We are using ice.
In synthesis bubbles.
Still you will take out. Still it doesn't maintain. See, now only I want to do many things. I want to check and all. This is not the way you do actually.
So I
We can't do now in cold room because you people are there, the camera thing and all. I'm just, this is a demo. You should not do like this. Jerome, this temperature sensor I'll keep just outside
In the there's a
the hall like this. So now we will remove the cell debris. So whatever now it is ruptured, membrane is there, cell wall is there, other organelles, everything. Now we will at least remove the larger cell debris just by centrifuging. How much we will keep? Like 5,000 maybe. So 5,000 I'll keep. Cell debris
still
removal
4 degrees required.
for 7. Fine.
3000 RPM for 5 minutes.
3,000 RPM.
I'll go for 3000 RPM 10 minutes. Very low. heart tubes. No, now after take the supernatant, sit.
ഇതെന്താ?
Otherwise, we'll reduce to 5 minutes. 3,000 now. I did
ഓക്കെ.
3 minutes only.
സ്റ്റോറിങ്ങും പിന്നെ ഒരു വാട്ട്
I did 3 minutes only. Maybe I'll make it 5. We'll stop it at 5.
യെസ്.
After 5 minutes, we'll stop
ആ
it.
സ്റ്റോറി. ഇത് പിന്നെ ഒരു സാധനം കൂടെ വേണം. 80% അസറ്റോണ് ഒരു 3 ട്യൂബില്. 995 മൈക്രോലിറ്റർ. 5 മൈക്രോലിറ്റർ ഞാൻ തൈലക്കോഡ് ആഡ് ചെയ്യും. മനസ്സിലായോ? ഇതുപോലത്തെ 3 ട്യൂബ്. അത് എന്നിട്ട് 4 ഡിഗ്രിയിൽ വെച്ചാൽ മതി. ഇത് 4 ഡിഗ്രിയിൽ തന്നെ വെച്ചോ. ഇതിന്റെ കൂട്ടത്തിൽ തന്നെ വെച്ചാൽ മതി. ഇതിന്റെ കൂട്ടത്തിൽ 3 കൂടെ. യെസ്. now
very
transfer
important.
this membrane part.
Yes. So.
YouTube.
So if you feel they are completely settling down and your supernatrants are white, that means sonication is not proper. You should repeat. 60%. It's there near my.
You can keep this for centrifuge. It's ready. Ragendu,
Same RPM. 6,000. How much? 6,000.
6,500.
Or just supernatant again.
No. So in lower RPM when you keep, the heavy particles will be settled. Now you increase to 7,000. So the whatever membrane, everything, it will settle now. That's it. The let go. What? No, I didn't add anything. No. Now we will get the pellet. Then we will add the next buffer.
Now pellet is required.
Now we need the pellet.
Before the simplification, what was the other simplification RPM? Say this
3,000.
is some special case, or normally only one certification is enough. This is a special case, or normally one certification is enough.
No, first centrifugation we did 3,000. So in 3,000 RPM, light particles won't settle. Only the heavy particles will get settled down. So you removed it. Now you will only pellet with more RPM when you keep. Obviously the membranes will get pellet.
The first 3,000 is the heavy particles.
Yes, that is a cell debris.
Today we are doing gel also. Gel.
No.
Told we do that fluorescence.
That we will do.
For that we should write the protocol.
I mean, in Japan we went for drinking.
That's fine.
So when you were speaking, it was off. But that time it was off. It was off. They kept it breaking.
Also, can you explain when we added the protein inhibitors actually?
Mm-mm.
So what will
Not
happen?
protein inhibitor, protease inhibitor we are adding.
Protease
Inhibitor.
Can you explain how this? What will happen?
Yeah, nothing is there. When you sonicate, everything is outside. Proteins, in a cell, everything is protected. The cells inside the protein, PS1, PS2, everything is protected from protease. But once you open this, the cells burst open, then proteases and proteins, everything is nearby. So you consider PS1 is there. There is some proteases which degrade protein, PS1. If it comes in contact, you are going to lose the PS1 structure, right? So you need to just stop or inhibit the proteases in the media or in the solution, whatever it is coming out. So we are just adding some inhibitors which will inhibit the proteases.
To protect the proteins.
Yes. So that our proteins are protected.
Yes.
Yeah. When you make a mixture of this all inhibitors, it is called cocktail. It is done. 3 minutes. Now we will do the brush. Then we add TRP2. Then again one more centrifuge. TRB2 one time only.
Now you did it at here.
No.
Same.
Yeah, two more will be there. TRB1
TRP2 is nothing.
for storage. Storage, yeah.
One only two types.
How does you want to take it by yourself? Sort of like this, but
will use the brush, right? Can you bring?
There is the
Check it out.
protocol after this.
6,500 we used 6,500.
Yeah. Now we will transfer
Change it
this to this tubes and add TRB1 with adding 1 ml initially on complete erase. Just
Pellet.
resuspend.
We'll
There
transfer
is
the
no,
pellet,
yeah.
Just cut the super.
Yes, but I think this starch also you don't need to take that white color. In that it is less. In this it is more. Yeah, let's take then with brush because starch is there.
First TRB. Anyway, remove
We'll
the.
remove.
In case you want to see the protocol.
Yes,
Why it not extra?
Now we are removing the.
because you see the pellet and you can see the starch content and all here. Because it's break open and you get all this, so you don't need. You need this green part. So use a brush and just take out that and transfer to some tubes like this. Because now we are going to pellet it in a very high RPM. So this will break.
TRB 1.
Yes, yes. This one. Ragendu.
Ragan is below the fridge.
Yes. Here there is MgCl2, CaCl2 and different concentration of sucrose.
That is just we are separating the complex.
This protocol we will give, no?
That will be complex. It will be put there in the suspension. This process will be complex. The buffers you use will different. First, we not get it. And the method of disrupting will change. But other than this, it's
Jerome, this one better we do without that. Maybe
without which.
the starch.
Okay. Should I?
Yes.
Just take this. Something added TRB1.
TRB1, next buffer. Now we were doing everything in CRB, right?
Now we are changing to TRB. TRB means thylakoid.
Yeah, I just kept the upper part like this and remaining I took. Okay, we can collect.
Just add 1 ml.
Yes, yes.
You can see the protocol side. It will be there. It will be having same like sucrose, everything.
To maintain the osmoticum, you are exposing all this to some media, so you need to maintain the osmotic balance. So sucrose is needed, and also it helps in stabilizing also. That's it. Yeah. Sorbitol also does the same function.
You can add, maybe make it up 20. But calcium is really good.
Calcium magnesium is part of membrane, right? Photosystems, calcium and magnesium itself present in the photosystems and all. So this will help to stabilize them.
Can I make up the order?
Yes, this also will do. But add little only we need. Like we get very little.
Right?
I think that is a little bit more. Maybe half. Yeah, add the same. No. Shall I add? No.
Can we stop here?
Fine.
Right? What? We can adjust.
You read and tell me tomorrow that one. What is EDTA function? Yes. Yes. But how it will help here? Tomorrow I will ask.
This multi-layer steps we detected. So the unwanted particles like debris we need only the complex protein complex. Apart from that,
Maximum 10 I will keep.
What is it?
You should keep like maximum, right?
Yeah.
But how much we can go for centrifuge?
What is mentioned?
It's the maximum 13,000,
Okay,
but
keep 10.
maybe 10.
10 for 10.
And then TRB2 and taking the things. That's it, right? We can keep 10,000 here, no? Recently any problem happened? No, right?
How can I use this particular reason?
I think so, but
Just for others, like
I understood, but I don't know for 77K how much needed. Just ask then Jyoti and check together.
ഭയങ്കര ഗ്രീൻ ആയിരുന്നു ഒന്നിനെ ഇതുപോലെ
അത് നമ്മൾ സോണിക്കേഷൻ ചെയ്തപ്പോൾ ഇടയ്ക്ക് ചില പ്രശ്നങ്ങളൊക്കെ വന്നില്ലേ, ചിലതിന് സൈക്കിൾ മാറിയൊക്കെ കൊടുത്തില്ലേ. നന്നായി ഡിസ്ക്രിപ്റ്റ് ആയിട്ട് എല്ലാം പുറത്തു വന്നാൽ നല്ലോണം മെംബ്രെയിൻ കിട്ടും. മര്യാദയ്ക്ക് ആയില്ലെങ്കിൽ അത് ഭയങ്കര ലൈറ്റ് ആയിരിക്കും.
എനിക്ക് ഇന്നലെ ഫസ്റ്റ്
ഇത് സെൽസും കുറവാണ്, കുറച്ചുകൂടെ സെൽസ് ഉണ്ടെങ്കിൽ കുറെ കൂടെ ഇവിടെ കിട്ടും. മനസ്സിലായോ? നിങ്ങൾ 1 മണിക്കൂർ 10% പൾസിൽ വെച്ചു, എന്നാ പിന്നെ പൾസ് കൂട്ടിക്കൂടെ? ഐ മീൻ ആംപ്ലിറ്റ്യൂഡ് കൂട്ടിക്കൂടായിരുന്നോ? 20
കൂടിയപ്പോൾ ടെമ്പറേച്ചർ വീണ്ടും
ഭയങ്കരമായിട്ട് കൂടി ഉണ്ടായില്ല.
അത് മറ്റേ
They will just form a bond.
ടെമ്പറേച്ചർ
4 ഡിഗ്രിയിൽ വെച്ചിട്ട് ഐസിൽ വെക്കണം.
Stable. That's why we use them.
സെൻസർ
So in
ഐസിൽ
the
വെക്കണം.
TRB1,
അങ്ങനെ
they
വെച്ചില്ല.
maintain the complex integrity. TRB1 purpose is that. Then finally TRB2 is for storage.
സാമ്പിളിന് ടെമ്പറേച്ചർ
Finally,
അറിയാൻ
and
വേണ്ടിട്ടാ.
right now we are using TRB1 only. After completely we isolate the thylakoids, we resuspend for storage
ഈ
for
1
later purpose.
അവറിലേക്ക് പോയതിന്റെ
In
അവസ്ഥ
minus
എന്താന്ന്
80 we'll store.
മനസ്സിലായില്ല.
So that time we use TRB2 buffer. A slight variations will be there. If you have gone through the protocol, there won't be a major magnesium will be there, triazine will be there, sucrose will be there. So all these are just to maintain the pH, and osmotic and pH should be maintained, as well as apart from that they should form a disulfide bridge. So that's the major point.
അപ്പോഴൊക്കെ സൂപ്പർനെറ്റിന്റെ
Yeah, that's what
താഴെ
these are all.
degrees ഒന്നും വന്നില്ല.
It's not like you cannot say one particular
Okay.
They are like well optimized. They have tried different methods and they found all of them together give a cumulative buffering effect. They can able to preserve the complex orientation. Our ultimate aim is to isolate a thylakoid complex, not an individual protein. Okay. Yeah, it's all a buffering. You just Google them. What are the individual components, and you will be identifying. These are all for any buffers. What are the components you use for any buffer?
അല്ല
EDTA,
നിങ്ങൾ ആദ്യം
salts
ഇപ്പൊ
you
ക്ലാമിന്റെ
use.
ഒരു പ്രോട്ടോകോൾ
So,
വെച്ചിട്ട്
to
എത്രയാണോ
maintain
വരുന്നത് അത്
EDPH,
വെച്ച് ചെയ്തിട്ട്
Western blot also
അതൊന്ന്
EDTA you will be using.
BNO എന്തെങ്കിലും നിങ്ങളൊന്ന് റൺ
These
ചെയ്ത്
are all
നോക്കണം
just like a
എന്നിട്ട്
buffering
വേണ്ടേ
agent
നമ്മളത്
to maintain
തീരുമാനിക്കാനായിട്ട്
osmotic.
പിന്നെ ഇതൊക്കെ ഓരോരുത്തരുടെ എക്സ്പീരിയൻസ് ഒക്കെ വെച്ച് മാറും. ഞാൻ നോക്കിക്കാണുന്ന രീതിയിലായിരിക്കില്ല യൂസഫ് നോക്കി.
Right here in this
നിനക്കൊരു
process.
2-3 വർഷത്തെ എക്സ്പീരിയൻസ്
This is
ആണോ?
direct answer is it's like well optimized already.
നിനക്ക്
So it should 90%. It should, if you follow the protocol everything properly, it should have the thylakoids only. And later on we'll go for finally thylakoid BN page. So we run in the gel. They will have different pore sizes, right, from 2 to 10%, if I'm not wrong. 2 to 12%, I guess, yeah. So in that the complexes, because every complex will not be same. Some will be very big complex, some will be small. So based on their molecular weight, they separate at different layer. So there you can confirm if your separation is very good, then you can get a clear distinct bands based on their molecular weight. And for Chlamydomonas, for well standard model organism, these bands have been well standardized already. Any paper if you go through, there will be like a standard band for thylakoid. These many bands will be there. There is a recorded. So you can cross check with your isolation with that. So if it's matching, then obviously your isolations are perfect.
So we'll know that if we have done anything wrong.
Yeah, based on the gel running, you can confirm. If you get a distinct band, then your preparations are very fine. That will be the most ideal one to confirm.
Can you use the stored samples also for the thylakoid isolation? So, for example,
Yeah, you can use it. You store it in the CRB buffer.
oh, not in nitrogen. After the dipping,
No.
in nitrogen,
Liquid nitrogen.
Nitrogen like
No, you wash once with the CRB buffer, CRB buffer,
Okay.
and then you remove the supernatant.
Okay.
And as usual, you store it in the liquid. You dip it in the liquid nitrogen and store them.
No, not I'm asking. What I'm asking actually, for example, higher plants.
Okay.
For example, if you take the millet,
Ah.
so I collect the sample, a root or leaf, whatever. Sorry, only we are doing the leaf only. Sorry.
Yeah, thylakoids.
Sorry. Leaf. We are collected the leaf sample.
Yeah.
Then can we store a few months or in?
Yeah, you can store because our lab member also do just that.
Okay.
They immediately collect the samples and they freeze in the -80 and they store.
Okay.
So whenever they want to do, they will isolate.
Okay.
It's not required that it has to be done on the same day only.
Generally, what we do is nitrogen we are using for this for store cryopreservation up to 6 months addition.
Yeah, that's what. Once you store it in the liquid nitrogen, you can keep it. Say it's like the cells will be completely. Everything will be stopped. The metabolic process,
metabolic
everything will
activities will be
it will be frozen at the particular time point what you are harvesting. So our lab members also who works on higher plants and all, they store.
So
Yeah,
same.
freezing. Huh? Yeah. Even we go for that, but for algal cells, we always advise to do fresh. If not possible, then it's okay. You can go. Nothing wrong in preserving. It is not going to change much. But when you are checking a very precise difference between different control versus treatment, it's advisable if you can do a fresh sample. But nothing wrong in keeping a stored one. But you store in the CRB buffer.
In small scale, we will
And in case if you are going for multiple sample, also difficult for you, no? You are storing the samples like 10-20 accumulated, and at once you are going to harvest 10 samples will be difficult isolating thylakoid.
Yes.
So you can isolate the thylakoids then and there, and keeping will be easier for you as well. Thylakoids also can be stored. So I prefer isolating thylakoid and storing will be more better way than storing the sample. But in case you feel
storage in that
TRB2 buffer. Anyway,
After
you are isolating thylakoid and storing, and in previous case you are storing the sample, and whenever you want you are isolating. Both are similar, but I prefer isolating thylakoid and storing. It's up to you, but I don't feel any major differences is going to come if you are going to also.
So if you want to do the replications,
Yeah.
So in plant sample, so it's very difficult to
same day
same
extraction.
day extraction.
Yeah, that's what I said. If it is not possible, you can do it. It's not going to affect a major. It's not a big problem. But store it in a proper buffer or proper way. That's very important. You should freeze the cells at the respective time points. If you are not properly store them,
Next step you are going for scan.
Sorry, sir. 77 speed. I think sir is very much interested in 77 Kelvin. We'll do it, sir. After I think 4:30 already. Maybe another 1 hour will take for completing this isolation. Maximum 1 hour. 40 to 1 hour will take for completing isolation. Once we are isolated, then we need to do chlorophyll estimation. Because equal chlorophyll, we need to do the using of this thylakoid sample.
Okay.
We need to estimate the chlorophyll, and in case if you have 5 to 6 samples, and every sample should have equal chlorophyll basis. For any experiment you go, either CD or 77 Kelvin, it should be equal chlorophyll.
So isolate by using of the isolated thylakoid.
We'll estimate the chlorophyll concentration per microliter, how much per ml, how much microgram of chlorophyll is present. We'll calculate, and based on that, we want like 3 to 4 microgram of chlorophyll for 77 Kelvin. We 70% ethanol is there, no? 70% acetone. Sorry, 70% or 80% acetone. Acetone. No, this is well standardized, sir. 80% acetone we directly use. No, nothing. It's a standardized protocol.
Jerome, so by using of the isolated thylakoid, so we can do different kinds of the experiments actually.
Yeah, thylakoid, once you isolated and kept n number of experiments you can done from.
Can you tell me some experiments?
77 Kelvin we are going to do. CD
77.
Yeah, 77 Kelvin. 77 Kelvin means low temperature fluorescence.
Low temperature
Fluorescence.
Okay.
CD you can do.
CD also.
Yeah, CD also you can do. And this one, ultra centrifugation you can do. BN you can do.
BN. What is CD?
CD circular dichroism. Circular dichroism. It's a spectroscopy which used to tell the pigment protein interaction. Means the thylakoid will have the pigments chlorophyll and carotenoids, right? So they will be telling how well the organization between the pigment and protein. Between you have 5 samples, and your control versus treatments will be in case of treatment definitely there will be a hampering, right? The pigment and protein got disturbed. So CD will tell you what is the intensity of disturbance by comparing the control versus the treatment.
So omics studies also we can do.
Anything you
By
can do,
using of the thylakoid or bands after the doing the BN page.
No. BN page will tell you different complexes. It's up to you how you want to further your study.
Okay.
If you want to, if you in case you found some special band which is not recorded earlier studies, then you'll be curious, right? What is this band specifically came in the treatment? Let's take in your treatment you got an extra band, whereas in control it is as usual. Whereas in treatment you got an extra band. You will repeat 1 or 2 times. Still you found the same. Then you might be interested, right? What is happening? Then you will need to identify. Then you will go in different way. You will go for mass spectrometry.
How many complexes you can see?
You can see there are 6 to 7 at least. Super complexes, PS1 complex, PS2 dimer. You can.
Sir, 77 Kelvin low temperature fluorescence, CD, EM, UoH, and Western.
Yeah.
And one more you told.
Ultra centrifugation. Those are. It's also a centrifugation. It separates different fractions. For very pure thylakoids you want to isolate,
What is the RPM
you can use that.
Out of it's what RPM?
I haven't done ultra centrifugation, sir. You can check the protocol out. It's a common protocols will be available.
High
I'm not sure. I cannot say directly without knowing. You can check it in the online. Very high speed, definitely. But yeah, you can check it in the online. Exactly, I haven't done it, so I cannot.
So ultrasonography, which instruments we can use?
Means there is in our facility also ultra certifications is there.
Okay.
It's a very big
So for
instrument.
which purpose you are using this?
To separate a pure thylakoid, fractions, different fractions will be there, no?
Okay.
Okay, as centrifuge, this ultra centrifuge different. Then I have to purify with affinity of column memory.
No need actually. They will be doing Western blot. After there will be different fractions. So already based on the Western blot, they will identify which fraction has which protein.
Okay.
So the study will go in different directions.
Understood.
Can we go for by using of the thylakoid, HPLC, such kind of?
HPLC, yeah, you can estimate the pigments, chlorophyll, carotenoids. Yeah, I think we have a schedule. One day we are going to do the Northeast. We have, no, we are doing on Thursday, I guess. We are going to do on Thursday.
But in our program,
It's not mentioned.
not mentioned.
Okay, I'll speak to. Maybe I'm not sure, but you can do it.
Which column you are
C18
the pigments in
partition through that. Different based on their retention time. Yeah.
class content, some other metabolites also only pigments.
No, in terms of HPLC, you need to be very focused. Which pigment you want to study? If you want to go for based on your standard, in case if you want to study chlorophyll, you will be checking based on the standard chlorophyll standard will be there. You will just compare with that. You won't study in HPLC. What is your aim? If your aim is chlorophyll, you will compare with the standard of the chlorophyll. What peak? Just check the intensity of the peak, and you will calculate the concentration. Simple as that.
If you want to focus on only chloroplast DNA. So by using with this thylakoid, isolated thylakoid,
You can, yeah, of course. If you are very specific to the
DNA isolation
chloroplast,
by using of this.
sorry,
By using of the thylakoid, so can we do the DNA isolation?
no, exactly. I'm not sure we can. I'm not sure about the method for specific isolation of the chloroplastic DNA, but transformation we can do for sure. Maybe I need to check.
Okay.
But there should be a possibility. But in this direction, I haven't think about it. But there should be some method. Definitely, there should be some method.
So in chloroplast, the RNA also will be there, I think. Is it there?
Chloroplast?
In chloroplast, RNA also present?
Yeah, of course. Everything will be transcribed to RNA, mRNA, and then it will be translated to protein. So I will be having a 3D nuclear structure. Tomorrow I will show you in my first presentation. So chloroplast will have. I think today also sir would have shown you. RNA isolation. Yeah, it's a global RNA. You cannot say contamination means it will be a global RNA. Yeah.
That means we can't say that's only RNA of chloroplast.
No, we cannot say. We cannot say. The trisal method, I don't think so. Any method available only for chloroplast RNA, I don't think so.
Then another tomorrow, I think.
Cloning, right? Cloning and transformation.
So, how can we be sure that transformation is occurring in chloroplast DNA?
Yeah. Tomorrow we are not doing exactly chloroplast transformation. We are doing nuclear transformation. Yeah, but chloroplast transformation, that's what my topic tomorrow for first hour is chloroplast transformation. I will be answering you this how to do it. So be there in the morning, 9 o'clock, 9:30, right?
Cloning.
Tomorrow practical is transformation. BN will be, I think, day after tomorrow. Yeah. We are doing nuclear transformation tomorrow. If tomorrow time is there, we'll do tomorrow, or day after tomorrow, sure. We'll extend little day after tomorrow. Because tomorrow is tightly packed. Let's see how fast we can come. I think only mall bio takes a long time.
What we're doing on Thursday?
Thursday, I think HPLC, I'm not sure. I need to check the product. Program mentioned something. Most probably HPLC.
In 77K today.
Check it.
77K one.
77 Kelvin is today. After this, we'll go for 77 Kelvin. Fluorescence kinetics?
Yeah, no.
Yeah.
छोटा वाट होता है वीडियो की सेकंड
Yeah, pigment is HPLC. So prepare well. There will be last day quizzes there.
Based on all the
Based on whatever taught only.
They will be offline.
tutorial as well as
Yeah.
lectures and tutorial.
Actually, quizmaster Venki sir, he's not there. He's only planning. So
It will be like a team.
mixed everything.
theory and theory.
Yeah. So what step is going on now? Still. Got it.
Are there softwares which are like conducive for reading the dates we can do experience?
Yeah, those things she will be telling you definitely on Thursday. Thursday she'll be taking class, no? Thursday she'll be telling you. No. Next program maybe. So you are following whatever happening here today? It's fine. It's little harsh, more complex, simple topic. Simple. Oh. Okay. I thought it's too complex. Oh, we are still. It's up to the audience. Let's see how you perform in the final exam.
Final exam here?
Quizzes. Now she's CQs. Everyone are making so. No, it's not a revenge. It's not a revenge. It's like a reflection to see, okay, how many percentage has it reached you. So that if any mistake from our side or anything need to be rectified, so that we can make changes in the next program. She has a lot of questions.
I'm asking like in Kakashi sir's class, those questions also come over.
So,
Oh, you heard.
No, it was break. So
So now almost we are at the end of the thyroid. Just ice the bottom.
also that acetone. Now you see, I told you you can use this thylakoid membrane for many experiments like Western blot to see the level of, if you're working with a stress condition and all, how D1 D2 is changing, how the LHC proteins are changing, which is increasing more, which is enhanced, which is decreased. This all you will compare. So you can do Western blot analysis, BN page, then this biophysical experiments like 77K CD. But for everything, I will tell you one example. Like if you are doing normal Western blot, most of you people must have done. So you keep a like equal protein you will load, right? Equal protein you will load. So in the same way, when you work with photo system, you need something to be to make it equal. Like you are going to check whether in the control and treated they are equal or what is happening. So you need to make equal photo systems or something equal you have to make. Either you can make it like equal protein, you can check the protein concentration in each and make the equal concentration. So every experiment, for example, you are going to do 77K, you will take 5 microgram of protein from the control and 5 microgram of protein from the treated one. So here normally we follow equal chlorophyll. Okay, so equal chlorophyll we are indirectly putting in a way like we are making equal number of photosystem we are going to compare. Understand? In a treated condition, the photosystems might be very less in treated condition in some cases. But you don't want to compare function of one photosystem to function of 100 photosystem in control. That is not the way you should check the efficiency or check the protein content. What you have to make, both make to 10 or both make to 100 and then check. So basically what we are going to do, we are making equal chlorophyll. I will check how much chlorophyll is in this, and if there is a treated condition, I will check what is the concentration chlorophyll present in that, and I'll make them equal. How we do a protein estimation, like what is that, BCA method or what is the other, Bradford method. So these are the method. You check the total protein in treated, what is the total protein, here what is the total protein, then you take equal amount of protein and you load. In Western blot and all you do this. So the same way you will do here equal chlorophyll. So before, once you done with your thylakoid, immediately do the chlorophyll concentration. Whatever, if you have 9 samples, 10 samples, do the chlorophyll estimation immediately. So here we use a acetone method normally. Just take a little amount of whatever thylakoid you have, 1 microliter or 5 microliter, mix with acetone, do a stirring and centrifuge it and go for the normal chlorophyll estimation. UV visible spectrophotometer, I hope you all know how to use it. It's a basic thing you learn in graduation itself. So take the chlorophyll estimation using acetone method and do it. Okay. Teach good. Okay. Now we will do, I will explain how we are going to do acetone method.
We can fraction for chlorophyll estimation, we need 10 micrometers per duplicates 30 micrometers and remaining all samples we will put in the
Who all did chlorophyll estimation earlier? Everybody must have done, right? Or anybody there? You didn't? Okay. We will explain.
It's a very simple.
I told you, we should be like very gently doing. That's why we are using a brush. When you use a like this and all, you are putting some pressure. So to avoid that, use this brush.
Very gentle. That's very important. Now onwards, you imagine that you are going to treat with the complex. If you put more pressure, there is a chance that they may get separate.
without tip you are using the brush.
Yes.
In acetone you are putting that one.
No, you should not. Acetone you put, then it is over. There is no membrane, nothing.
Okay.
Acetone will take only the pigments and remaining things will be
To be separate.
thrown.
This is buffer only.
This buffer. You are going to store it in -80 for all your experiments now. And I'll take a little for chlorophyll estimation. What acetone will do? Just take the
Just following the protocol. Because
Anybody wants to do the other tube? Come. Slowly do. There should not be any clumps or anything. Take out all the thing and keep in ice also. Because we will use this to. I added this. Yes.
How much volume and or little
You should be like standardize this. Finally, you should get a concentration per microliter of whatever you have should be 2 to 4 microgram of chlorophyll should be there. So now I know if I see the pellet, I know if you add how much I will get it and all. I have an idea. But once you start doing this, you will understand that. And once you standardize, every time you do the same. Still you will see little bit, but maximum is removed. No clump should be there, everything should be homogenized. Do like slowly, let it get completely.
Ma'am, can you once just show?
Oh, sorry. It's done. We can add a little bit more. Can you open this one? So I'll pool both into one because now we don't have treated and all, we are doing only control. So I'll have only one sample. Jerome, it's done. Maybe, yeah, I'll do the chlorophyll part. You can make the aliquots and freeze.
okay, I'll make it 100.
Yeah. 100. 10 microliter pipette. This you can use for.
So
And
all are for same labeled.
This is only for acetone. Those others.
Labeled,
No label.
but where box you have some common box.
I don't have.
-80 you should treat, no?
-80. Just maybe for only
Tomorrow?
this 2-4 days, right? You can wrap it in aluminium foil and keep. You can use this. This has some problem. This side is not there. This one.
So everything
So-
is pulled in on, right?
Yeah. I'll take for acetone. So I'm taking just whatever we got. See, this much only we got. So I'm going to take 5 microliter for acetone, like chlorophyll estimation, and the remaining we will using liquid nitrogen we will freeze and keep it in -80, and later we will use for whatever the purpose we have. No, day after tomorrow. I'll take only 5 microliter for this estimation.
Breathing is same as dropping.
Yes. Acetone I am adding 995 microliter of acetone and 5 microliter of thylakoid. No. Whatever in thylakoid membrane, whatever the pigments will be present, you will get all. Acetone method, chlorophyll. Acetone.
No, first I'm taking 5 microliter of extraction.
Yeah, but we are going to give a specific wavelength, right? For chlorophyll. Extraction, it will do all the pigments.
Spectrum.
Yeah. Yes.
This pigment extraction is based on frequency.
Which pigment extraction?
When we add acetone,
This is acetone. Once you add organic solvent, membranes will dissolve and all the pigments come out in organic. Right? Estimate you there. Where is Jerome? Jerome, it's done. You can do this. I'll do. ഒരു വട്ടം ഇത് ചെയ്യണം സ്പിൻ ഇത് രണ്ട് ജസ്റ്റ് ഇത് എടുക്കുക ബ്ലാങ്കും വെക്കണം സെയിം അസറ്റോൺ എടുത്തോ So I think I don't have to explain the acetone part, right? Chlorophyll estimation. So I just mixed 5 microliter plus 995 acetone. Just one spin it and remove the whatever membrane particle lipid you will get. So spin one, like 5 minutes or something and take the entire 1 ml from here and go for UV. You will take, we follow Pora et al method, which is like 645 and 663 nanometer. You will take the reading. Both you will take the reading. Yeah, this is the chlorophyll absorbance we are taking, and using this whatever I mentioned, that method we have a particular formula. If you get the 645 nanometer and 665 nanometer, we put this into the formula and you get the total chlorophyll from that. This part you already know. I will explain you. So write 5 microliter plus 995, this one, and just mix it properly. We have a small spinner centrifuge here. Yes.
You can see the both scan and as absorbance both.
Absorbance.
Both scan as well as complete evidence scan. 200 to 700.
We can do that also. Absorbance spectra. Yeah.
First time actually we are interested. Yes, sir.
Okay. We'll do. The thing is 77K itself will take some time. So we'll see, we can
So here, you said.
where? Yeah. No, this is not okay. It is coming. This is working, right? This one? Yeah. Switch on?
No?
Yeah.
Okay.
Now it's okay. My phone? Just wait. Hello? Hello. How are you feeling? Having fever? What? Lab only, work